All right. Well, good morning, everyone, and welcome to the 2023 Northeast Regional Invasive Species and Climate Change Management Network Symposium. We're super excited to have you here. My name is Bethany Bradley. I sit in Western Massachusetts at the University of Massachusetts Amherst, and I am opening this session on behalf of the Northeast Risk Leadership Team, which is about 25 people that you'll see or hopefully not see some of them behind the scenes today. So just an appreciation for all the folks who are making this happen. Before I talk, so I have a little opening talk of some of the things that we've been up to over the last year in the RISC network. Before we get into that, so a little bit of housekeeping. The first is that we are trying a new experiment today and tomorrow where we're keeping everybody in a big Zoom meeting rather than a webinar format. And the reason for that is we want to make sure that everybody can participate and feels part of the conversation. But that also means that it's going to be easy for you to unmute and be heard. So if you're not speaking, please just keep an eye on your microphone to make sure that you're muted. We are also recording this presentation and we'll be posting the presentations and panel discussions to the RISC website later. So if your video is on, there is a chance that you may be recorded. So just bear in mind that if you want to make sure that you're not recorded, it's totally fine to keep your video off. We also have closed captioning enabled in this Zoom meeting. So if you'd like to see closed captions, the way to do it is at the bottom of your Zoom screen, you should see a More button with 3 dots above it. If you click on that, there's a captions option, and then you should be able to show captions. You can also change the language, although I don't know what happens if you try to caption me in French or some other language in there. And if the captioning is annoying you, you can do that same process to turn the captions off. In terms of our last piece of housekeeping is we want to make sure that this is open, accessible presentations that all of the speakers and panelists today and tomorrow are looking forward to talking to you and hearing from you and want to know what you have in mind and your questions. You are most welcome to use the chat, which you can see sort of in the middle of of your Zoom bar, to exchange information during the presentations. So the folks who are speaking may not be actively monitoring the chat, but the risk leadership team and everybody else is looking through the chat. So you're welcome to have those sort of sidebar conversations to discuss and ask questions during that time. At the end of presentation, so during the question and answer period, the moderators will be moderating between questions that are posted in the chat and raised hands. So probably the best way to get a question answered is to raise your hand. Zoom does preserve the order in which hands are raised. So just keep your hand raised until we get to you. All right, so again, just a big acknowledgement that there are a lot of folks behind the scenes making this event happen. So these are pictures from everybody in the risk leadership team, and I added a Valentine candy because happy Valentine's Day, everybody. So much love to the risk leadership team for helping this and for making this happen. And especially just to Jenica Allen and Justin Dalaba, who have really spearheaded all of this effort. So I wanted to talk for a few minutes before I introduce our plenary speaker about what Northeast Risk has been up to over this last year. And I like to start with our mission statement. So our mission statement aims to reduce the compounding effects of invasive species and climate change by synthesizing relevant science, sharing the needs and knowledge of managers, building stronger scientist-manager communities, and conducting priority research. So I just want to go through those four things: research synthesis, sharing information, building networks, and conducting research, to tell you a little bit about all of the things that we've, maybe not all, many of the things that we've been doing over this last year to work on those four aspects. Um, and I will say that risk is always a work in progress, and we're here for you, for everybody in the audience. And so the more feedback we get, the more information we get about your needs, what's working, what's not working, the more we can better serve you and address your questions. So in terms of that first piece, synthesizing relevant science, this year we had kind of a banner year in terms of our management challenges. So those are the research synthesis documents. About a year ago, so last year in the spring semester, we had a management challenge on marine mischief. So looking at interactions between salt marshes, climate change, and invasive species. And then later in the year, a management challenge on sleeper invasive plants. So looking at invasive species that might be established, but not yet invasive in the Northeast region. And then last fall, we had a longer than 2-page synthesis document called Do Not Sell, which is an extended list of about 15 species that we want to try to avoid in the Northeast of selling as ornamental plants, because they're likely to expand their ranges into the Northeast with climate change. And one thing that we've added this year is a series of what we call coffee talks. So for each of those management challenges, we've also had coffee talks to give us a chance to talk about those syntheses and hear more about, how they could be used and how we could better deliver that information. The other big piece in terms of synthesizing relevant science is that we have now a pretty substantive archive of research summaries that you can find on the website. There's a search bar at the top, so this was just the first return. There were a bunch of them when I searched for marine, which happened to return a research summary that Cascade, or a research summary of a paper that Cascade led. So I thought that was appropriate given our plenary speaker. We have more than 200 research summaries on the website now, so please feel free to use that as a resource. In terms of sharing needs and knowledge, so one thing that we have focused on over the last couple of years is to put together conversations between a researcher and a manager on specific species that are expanding into the Northeast. And so this year we had a conversation about kudzu, definitely one that folks down in the Southeast, like the two speakers in this webinar, know well, and those of us in the Northeast are increasingly aware of. In terms of sharing needs and knowledge, we also have had some events like this pop-up lunch chat on hemlock woolly adelgid and climate change that we did over the summer. So opportunities to talk about specific species that that are a concern across different climate zones, certainly across borders, and share that information. Also, in terms of sharing needs specifically, so this spring, we will be sending out, probably within the next month or so, a new survey to gather information needs on invasive species and climate change. Some of you may remember 5 years ago, we did an earlier version of this survey, which we published, and we have been using that information, so what you told us 5 years ago, to prioritize the type of research and the type of synthesis that we're doing. And so we figured 5 years in, now is a good time to refresh that survey. So please keep an eye out if you're hopefully you're all part of the risk listserv. We'll be sending that out probably in March. In terms of building stronger networks, so we have been working also to put together discussion sections, sort of informal ones that are not themed across a specific species, but opportunities to just talk and meet people across borders who might be might form part of your larger network and be important resources in the future. We've also been supporting what we call the Invasive Plant Council's working group. So that's a working group that meets 1 or 2 times a year and has met for the last 3 years. And at this point, we're spanning from Pennsylvania up to New Brunswick and Nova Scotia. So we've got representatives from all of those different states engaging and talking to each other a couple of times a year, just opportunities to exchange information across borders. And then the last piece of what we do is conducting priority research. So this is an example of one of the science papers that came out last year. And this one was in direct result of that Invasive Plant Council's working group. So one of the things that that was asked in that working group was, what's the current status of invasive plant regulations in the Northeast and invasive plant risk assessments across the Northeast? And so we did an analysis of that. And if you're interested in a database that has all of the results from state invasive plant risk assessments, this paper has that database appended to it. And it is, fortunately, this is an open access journal, so open to anybody. Another research paper that was published this year out of the RISC team is this one with so Bianca Lopez and Janica Allen led, which looked at interactions between invasive species and climate change. And one of the optimistic, the silver lining results from this is that we came at this thinking that if you add invasive species and climate change together, then maybe most of the time you're going to get worse results than you would expect by the two independently. It turns out when you add the two together, actually, more often than not, you get not as bad as you would expect based on the sum of those two effects. So a little bit of silver lining that if we can deal with biological invasions, then that goes a long way to addressing and building resilience to climate change. And then a last priority research area was us taking all of the information that we've learned from you and what we've gathered and put together over the years through the RISC network to talk more about how invasive species policy needs to integrate and embrace climate change. So if you're looking for sort of a synthesis of all of our thinking over the last several years, this paper is a good source for that. And the previous two, there are research summaries for those. There's actually a research summary for this one too that's you'll see in your inboxes in March. So just before I introduce our speaker for today, to go over the agenda. So in a couple of minutes, Cascade Sorte will give our plenary. We'll have a short break after that. There's a networking lunch at 12:30, so half an hour, and those have various themes associated with them. So if you hang out in the Zoom room, you can go talk to everybody else who's interested in some of those topics. And Cascade will also be in one of those Zoom rooms. So you've got an opportunity to engage more with her and ask any questions that we are not able to get to during the question and answer period. Right after the networking lunch, we have presentations about early detection tools and techniques. Short break after that. Then we are finishing up with a panel discussing building habitat resiliency. And then we're closing out with an exciting Community Action Award and some closing remarks from Tony Lynn Morelli. All right. I'm going to stop sharing my screen so that Cascade can go ahead and get her screen sharing going while I introduce her. So I'm super excited to introduce Dr. Cascade Sorte. So Cascade earned her master's degree at the University of California in Santa Barbara and her PhD at the University of California, Davis. After that, so that might sound like she's only a California person, but after that she was in Massachusetts. So she did postdoctoral research at the University of Massachusetts, Boston, and was in the Northeast for quite a while. In 2014, she joined the faculty of the University of California, Irvine in evolutionary ecology and evolutionary biology. And in 2017, she was elected as an early career fellow for the Ecological Society of America, which is a really impressive award. Cascade is an integrative marine ecologist, and her research spans ecological scales from physiology to biogeography, and she investigates the impacts of global change, particularly interactions between climate change and species invasions. And what all of that doesn't tell you is that Cascade is also a fantastic collaborator and one of those people who brings other people together and creates more opportunities for great research and great synthesis. So with that, I will turn it over to Cascade.
All right, good morning, everyone. Can you hear me okay? I'm up? Okay, good. Let me just get my my screen organized how I want it here. Okay. Well, it's great to be joining you remotely. I saw on the introductions that there are people from Hawaii. There's at least one person from Africa. So I'm really today appreciating that we can meet in this way remotely. I'm joining you from the University of California, Irvine, which is in Southern California. Sorry, I'm still, okay, there we go. As Bethany said, I've been here at UC Irvine for about 9 years now, before which I was in the Boston area. I'm a marine ecologist, so you'll see that a lot of the examples that I give during my talk today come from marine systems, but I've also worked quite a bit with terrestrial and freshwater ecologists doing syntheses related to climate and invasions. And today I'll talk about climate and invasions, but also evasions and expansions. Oh my. So I'm hoping today to have some new information for everyone. I want to start a little bit from the beginning about why we're talking about climate and invasions together, and then build up to talking about what I think are going to be some emerging new reasons that we're concerned about this interaction potentially. So both climate change and invasions individually threaten biodiversity, but they could also interact. And these interactions among the causes of biodiversity change represent one of the largest uncertainties in projections of future biodiversity change. And this was recognized back in 2000. So it's not a new idea, but we're still trying to figure out what this means as invasion biologists and global change biologists. And I just wanted to let you know that for my talk today, I'll be using the term invasion or invasive species to refer to any non-native species that has established outside of its native range. And I do that because in the system that I study, a lot of the non-native species are not well studied, so we don't have a good sense of their impacts. So I think compared to the plant world, there's less of a division between simply introduced species and noxious invasive species. So I'll be using the term invasion very broadly today. So what does climate change have to do with invasions anyways? Well, there are several ways that climate change might interact with invasive species, and I'll go through some of those and give some examples. So first of all, climate change could actually increase the transport of invasive species around the globe. And in marine systems, kind of a large or drastic cause of that, for example, could be that as the Arctic becomes increasingly ice-free, this could lead to increased shipping across the Arctic and moving organisms around in that region. And for example, some of my work I do up in Alaska, that's an area that has typically seen low levels of marine invasion, but we expect that the level of invasion could be increasing in that area as we see these shifts in climate. So I'm not going to talk a lot about this particular mode of interaction between climate change and invasive species today, but I wanted to mention it as a mode that could cause increased introductions. Climate change could also lead to disproportionate responses of invasive and native species. And this is an area that I've spent a lot of time doing research, and I found that responses to climate change differ between invasive and native species. So why might that be? Well, the same characteristics that allow success under climate change are characteristics that can allow success or high impacts as invaders. One of these is the tolerance of high temperature and broad temperature ranges, making these invasive species predisposed to do better as our world gets warmer. And so I have some evidence for this from the marine fouling community, which is where I did my doctoral research. And for those of you not familiar with the marine fouling community, this is a community of species that live attached to or foul hard surfaces. This includes natural rocky reefs, but it also includes a lot of human infrastructure, including boats, docks, pilings, etc. So this is an example of a picture of the marine fouling community. Those squishy things that you're seeing in that picture, the orange and yellow, are tunicates or sea squirts. And the purple one there is a species of bryozoan that's invasive in the United States, Bugula naratina. So I did some work in the fouling community and found that the temperature that these species can handle differs between the native species in the community and the invasive species. So if you look at the temperature that causes 50% mortality, the invasive species can tolerate a higher temperature before they start to die than the native species can. And this gives them a leg up during climate change. There's also evidence that the species in this community tolerate different temperature ranges. So this is work that was led by a master's student that worked with me, Robin Zarabecki, and she found that if you look at those same invasive species, they're occurring over a broader temperature range globally than the native species are. So they just innately have the physiology that will allow them to survive under a broader range of temperatures or survive under increasing ocean warming compared to the native species. Another reason that invasive and native species might respond differently and invasive species might be predisposed to do better under climate change is that invasive species in the systems I've studied appear to be more resistant to disturbance. So also in the fouling community, I did an experiment where I subjected the species in an established community, like on this little 10 by 10 centimeter plastic plate. I subjected them to heat waves. And I found that after the heat wave, there was a decrease in species richness or diversity on those in those experimental units on those plates, where starting out, there were about 6 species in the community, and afterwards there were only 2 left that were able to survive the heat wave. But even more importantly, is the proportion of that community that was composed of invasive species. So before the heat wave, there was about 60–65% of that community that was invasive species. But after the heat wave, the community was composed of 95–100% of invasive species. And that's because those invasive species were able to tolerate the heat wave where the native species weren't. And then a third reason that we might see disproportionate responses of native and invasive species is that invasive species are really good at capitalizing on increased resources. So to the degree that I think I got muted too. To the degree that climate change is increasing resources, that appears to benefit invasive species over native species. So let me just talk through this figure really quickly. Honestly, if you're not someone that likes to look at figures, you don't have to, but I thought you might be interested that this comes from a study that I did. As Bethany mentioned, I organized some synthesis groups. And in this synthesis group, we looked at studies that had evaluated the effects of climate change on both invasive and native species at the same time. And we studied work that was done in both aquatic systems, so that would be the AQ here, and terrestrial systems, the TERR. We looked at the effects, or studies that had looked at the effects of warming, increased temperature, increases in carbon dioxide, and changes in precipitation, either increases or decreases in precipitation. And whenever the data point here is above 0, that means that the invasives are responding more positively to that aspect of climate change. And we found that the invasive species were always favored whenever resources were increased, whenever there was more carbon dioxide, more precipitation, more temperature, which has been called a resource in aquatic systems. But the times when the natives responded more positively or the invasive species were not favored were the times when resources went down. So that would be increased temperature in terrestrial systems, which leads to water stress and decreases in precipitation. So these are three mechanisms or three reasons that invasive species might be predisposed to do better than native species under climate change. So to summarize this point, invasive species appear to respond less negatively to climate change than natives, but I just want to point out that I've made these little acorns here, which are our invasive and our native species. And the invasive species here is not smiling. They just have kind of like a ho-hum face, whereas the native species is frowning. And I did that purposely because I wanted to emphasize that this work that I've done and collaborated with others on is not showing that invasive species are necessarily benefiting by climate change. They're not necessarily getting bigger. Their populations are not necessarily growing, but they're not doing as poorly as the native species. So a third way that climate change could affect invasive species is that it could alter the impact of invasive species on native species. And this is an area that I've been really interested in more recently and where most of my research on this topic has been focused. And Bethany kind of alluded to this when she was talking about a recent paper, which I'm actually going to talk about in more detail now. So just to give you a schematic of how this might work, why might climate change affect the impacts of invaders on native species? Let's look at this example from marine systems in the Northeast. So this is our native dog whelk, which is Nucella lapillus. Nucella lapillus has been negatively impacted by climate change based on some work that I did that was published in 2017 in Global Change Biology. And at the same time, we know that increases in the invasive green crab in coastal ecosystems in the Northeast have had negative impacts on the native dog whelk because invasive green crabs like to eat dog whelks. And so these are both direct effects, direct negative effects that are causing declines in native dog whelk populations. But there's also the possibility, and some research suggests that this is happening and will continue to happen, that climate change could increase the numbers or the size or the feeding rate of the invasive green crab. And by doing that, there's going to be an indirect negative effect on the native dog whelk, meaning if climate change increases green crabs, then increases in green crab are going to lead to an increase in this negative effect on dog whelks. So this is an indirect negative effect. And what that does is it just compounds all of these negative effects on this native species. So this is an example of how climate change and invasions can interact to amplify the impacts of the one on the other. But there's also examples where climate change and invasions can interact to minimize impacts. This is one example where invasion-mediated effects on marine trophic interactions led to positive feedbacks and favored kelp persistence, where an invasive species was actually benefiting a native species under climate change. And then there are cases where climate change and invasions don't necessarily interact. So to show you this graphically in another form, if we have a native response, like say we're most interested in survival of a population of vulnerable species, then under control conditions without climate change and invasions, we have a survival that's around 100%. And let's say invasions are causing that survival to drop to 80%, so they're having a 20% effect on survival. And separately, climate change is having a 30% effect on survival. So it's decreasing survival to about 70%. Well, here's how these three options of how the interaction between them might go. So the first option is that there's no interaction between them, that you have a 20% decrease due to invasions, a 30% decrease due to climate change, and together that leads to a 50% decrease, right? That would be, they're not interacting, invasion and climate change not affecting each other, they're just layering on top of each other. But it's also possible you could have an amplifying effect where the survival, when you have invasions and climate change together, is even worse, right? You have 80% decrease in survival, and now you only have 20% survival of that population of interest. Or you can have this minimizing effect, where the outcome under both invasion and climate change is better than you would expect under those two separately. So looking at these different possibilities, this was a question that we set out to address as a working group. So you'll see some familiar faces in this working group. Here's Bethany Bradley, who did the introduction, one of the leaders of RISC. Here's another RISC leader, Jenica Allen, who was really the main leader of this study that I'm going to be talking about, along with Bianca Lopez. So again, to address this question of how climate change and invasions might interact to change the impacts of invasive species, we did a synthesis where we looked at all studies that had looked at the effect on a native response, such as survival, under conditions where you just had invasions, you just had climate change, and then under conditions where you had both of them together. And here are some results from that study. So Bethany gave a little teaser about this, and I'll go just a little bit more in depth. And I do encourage you to check out the study. This was the product of, I guess, about 7 years of work. So it took us a while to get this together, but the results are really, really interesting. The first result is we looked at just overall, what was the effect of the global change? We call it global environmental change here because in addition to climate change, we also looked at the effects of increased nitrogen. Then the effect of invasion by itself, which is this bar in the middle, and then invasions and global environmental change together. And this is the mean effect size, just showing how much of an effect was there. You can see the effects were all negative here. That would mean that each of these was doing things like decreasing survival or decreasing population size. And overall, what you'll see here is that the effect of invasive species, which is this black bar in the middle, was stronger than the effect of global environmental change, just separately. So this supports some other work that's showing that invasive species are more of a problem locally than climate change, at least on the scales that we are studying them right now. Also, interestingly, the combined impact, just generally, of invasions and global environmental change was not different from invasions alone. Again, the invasions are really driving these negative impacts here, and layering on the climate change in a broad sense was not increasing those impacts. Now, let me just show you that there's a lot of variability here, because you might be saying, well, why am I at this symposium and why am I interested in this topic if it's just all about invasions and climate change isn't changing the effect of invasions? Well, in most cases, in 67% or 2/3 of cases, we did find these minimizing effects, which would be this light blue and this light pink or red here. So those are the cases where together climate change and invasions are having less of an impact than they would separately. We found that 4% of the effects are additive. It's pretty hard to have an additive effect just because they would have to be sort of exact. But here are the ones that I think we want to remember and be concerned about. So I would keep the silver lining in mind, but also note that in about 1/3 of cases, 29% of cases, we do have an amplifying effect where the responses are stronger in the presence of both invasions and climate change than they are separately. So, what this means is that we want to look at our individual cases that we're interested in, that we're managing, that we're working on policy for, and try to figure out whether they are in these, minimizing parts of the bar or in these concerning amplifying parts. So to summarize this work so far on the effect of climate change on the impacts of invasive species on native species, we've found that climate change more often minimizes the impacts of invasive species, but it increases them in over 25% of cases. So this is a continuing area of study. We still have relatively few studies to pull from on this topic. And so I think you can expect to see more from our group looking at how climate change affects invader impacts. So now I want to transition to the second part of the top title, which was, this isn't just a talk about invasions, but it's also about evasions and expansions. Oh my. So another way that climate change can interact with invasive species is actually to drive a new form of invasions. And in this case, I'm calling these range expansions. They've also been called climate invasions. So this is the case where climate change is driving native species to shift their ranges and to spread. And when they do that, there's a lot of analogies, a lot of similarities between the spread of these native species and the spread of non-native invasive species. And so one of the topics that I'm going to focus on here is whether or not these native range expansions can have impacts as they spread outside of their historical range. So this is a topic I've been interested in a while. This is the study that Bethany pointed out came up when you searched the research summaries on the RISC webpage. And so you can check out the research summary there, which was written by a PhD student in my lab. And this is a study in which we did a meta-analysis, again, a synthesis of, in this case, marine range expansions. This was published over 10 years ago. So at that time, there was relatively little data on the impacts of marine range expansions. And at that time, I only found, 8 examples of studies of impacts. It did span a range of species. As you can see here, we have some disease-causing organisms. We have some herbivores like these limpets and these sea urchins, up to species like this giant predatory squid. And what I did find in this study was that these impacts of these native range expanders were the same or greater magnitude as the impacts of non-native invasive species. And to do this, I took each of these range expanding species and I looked for similar non-native invasive species and compared their impacts between the expander and the invaders. So for example, if I was looking at these predatory squid, I would be looking at non-native predatory fast-moving, large-bodied species, that sort of thing. And when I was looking at the effect of these gulls on nitrogen inputs, I would look at invasive bird impacts on nitrogen inputs. And the outcome of that study was, like I said, that these impacts were really similar between the native range expanders and the non-native invasive species. So this suggests that yes, these expanding species can have impacts that are just as concerning as the impacts of the non-native invasive species. But what if these previous studies have focused on the worst case scenario? This is really a case where we could see some publication bias, where people might just be publishing on the really problematic expanding species because they're really obvious. And again, there were only 8 of them that I found impact studies for. So I've been really interested in whether we can predict the impacts of these range expansions, but also interested in whether or not we can predict the impacts of non-native species invasions at the same time. So to think about how to address this question, I turned to this risk assessment approach that is called ICAT. It stands for the Environmental Impact Classification of Alien Taxa. And I think you've heard a little bit about it at RISC because Bethany Bradley has also been using ICAT in some of their terrestrial work. So I'm just going to give a short summary here. This risk assessment protocol is now the IUCN standard for classifying the impact of alien species on the environment. So the IUCN now is actually logging and recording and creating a database of ICAT scores for different non-native invasive species. And this work with ICAT, I have benefited from an interaction with an international working group, which is pictured here. And a lot of the individuals pictured here were actually part of putting together and adopting this IUCN protocol. So I want to acknowledge them. So how does this work? Well, the ICAT compiles all previous studies of a species of interest. Like if you were looking at the invasive green crab, you would do literature searches. You'd want to look in all types of literature. You'd want to look in peer-reviewed literature. You'd want to look in reports and compile all previous studies of the effects of the species of interest, say the invasive green crab. And then you would assign impact scores based on the level of impact. And this is a semi-quantitative metric. And so you'll see there are numbers related to these categories, but there's not an equal difference or distance between the categories, which you'll see when I describe what these categories refer to. So for example, level 1 here, minimal concern, which I didn't put a summary for, means that there's some evidence that this species may have impact, but it hasn't yet been measured. A minor impact is an impact level 2, and this is where your invasive species, like the invasive green crab, has caused a decline in the condition of a native species. So condition would be something like body size. Then a moderate impact is an impact level 3. This would be where your invasive species of interest led to a decline in the population size of the native species. And then level 4 is a local reversible extinction of a native species. So maybe the invasive green crab has caused the native dog whelk to disappear from a coastal intertidal site, even though we know that there's populations living, subtidally or areas where the green crab can't reach that could likely reestablish and repopulate if the green crab was removed. And then the largest impact is a local irreversible extinction of a native species. So if that invasive species is driving a native species to extinction to a population size of 0, and the native species cannot come back even if the invader is removed. So these are the different categories. As you can see, it's semi-quantitative because there are numbers on this. And as you'll see in a minute, I do some analyses where I use these numbers in statistical analyses, but you can also see that it's not really a continuous metric. So what I did in collaboration with a postdoc in my lab is I wanted to use this ICAT approach to look at the impacts of range expansions. And this is the first time it had been used in this way for native species that are shifting their ranges under climate change. To do this, we made a couple of changes to the way the ICAT protocol is described for use with non-native invasive species. First of all, it's only described for use with negative impacts, but I was really interested in looking at the positive impacts as well, especially considering that in this case, we're talking about native species shifting their ranges. But it is something that is increasingly being used with invasive species, non-native invasive species as well. So we looked at both negative and positive impacts. And to look at the positive impacts, we essentially just reversed the definition of the impact for the negative impact. So for example, if a moderate impact is a negative impact is a decrease in the native population size, then a positive impact is an increase in the native population size. And the other change that I made for this study was that I used all of the studies on the species of interest, including those from its native range. So typically ICAT is looking at just the negative effects in the invaded range. In this case, I looked at negative and positive effects in both the expanded range of the range expanding species and their native range. And that's because I'm really interested in whether we can predict impacts before a species has even spread. So one of the concerns about ICAT in general for both non-native invaders and for range expanders is that there might not be enough data, because the amount of data that's existing in the literature determines whether or not you can give an impact score to a species. So we studied 39 marine range expanders in North America. That was our geographic range. And we found that there were published studies for over 80% of the species. And so we actually were able to give an impact score to quite a few of the species. And that's what you'll see here. In the pink or the red is the negative impacts, and in the blue is the positive impacts. And you can see that impact score goes from an impact of 0, which none of them have an impact of 0 because that's not defined. But you can see the lowest impact is impact of 1. I've shaded that out just because an impact of 1 means there may be an impact, but it hasn't been measured yet. So I would mostly draw your attention to the impacts of 2 or higher, both in the negative and the positive direction. And we found that for those species that we could determine an impact for, half of those had impacts, had a score of 2 or greater. And these impacts included impacts on growth, which would be that impact level 2, abundance, which would be impact level 3, and population viability, meaning can a population persist in an area. We didn't have any impacts of level 5, which was that massive impact. So in no case could we really, confidently say that an expanding species had driven a native species to extinction where the native species could never come back. Although we did have some level 4, where we had, say, species that would graze down a native species to the point where it was functionally extinct. But if that grazer left, it was able to reestablish. So you can see that most of these impacts are negative. There is a little bit more red on this figure than there is blue, but about half of these had both negative and positive impacts. An example of that would be the red mangrove. This is a species that had negative impacts because it provided substrate that non-native invasive species could colonize, and so it actually increased the invasion of non-native species, which was considered a negative impact. But it also had positive impacts because it provided habitat for native parrotfish and also for some fisheries species. So that would be an example where there's a species that's having both negative and positive impacts. And looking at this question of whether we can assess impacts before species have even expanded, to look at this, I've plotted here the impacts in the species' native range and the impacts in its expanded range. So we had about 7 species that we were able to give an ICAT impact score in both their native range and their expanded range. And if those impacts fall along sort of a 1-to-1 line, like I've shown here in red, that suggests that the impacts are very similar in the native range as they are in the expanded range, which means that you could use the impacts in the native range to anticipate what the impacts would be in the expanded range. And we found there really was a strong correlation between that. And that's really heartening. It suggests that the impacts from studies in the native range are indicators of the impacts in the new expanded range. Now, if anything, impacts in the expanded range tended to be a little bit stronger than impacts in the native range, which follows the pattern with non-native invasive species as well. So what's next? Well, I'm really interested in the possibility of using frameworks that have been developed in invasion biology to predict the impacts of species range expansions. And so my work right now is focused on whether or not I can use impacts in the, native range to understand or predict impacts in the expanded range. So just looking at using like a risk assessment approach, like I just showed you. Also interested in using abundance, which is a pretty good indicator of impact based on some other synthetic work that I'm not talking about today. And also trophic level is a good indicator of impact because species at higher trophic levels tend to have higher impacts, especially early on in an invasion and perhaps also in a species range expansion. So the work that my lab is doing right now, our experimental work, is focused on these 2 predatory whelk species. On the bottom is the black unicorn whelk, Mexicanthina lugubrious, which is shifting its range from Baja California, Mexico, up into Southern California. And on the top there is the spire dog whelk, Acanthonucella spirata. This is a related species that's shifting its range in Northern California. Both of these prey on shellfish, including barnacles and mussels. And so we're interested to see how they're affecting other prey species and potentially competitors. And so these are just some pictures of our group doing work down in Baja, where it's really interesting. This is the black unicorn whelk that occurs in really, really high abundances, not too far south of where I live here in Southern California. So if this whelk achieved those abundances in California, I anticipate it could have a really strong effect on our shellfish populations, which are already in decline because of a lot of reasons, including high direct human impacts in our very urban area. So this work has been written up in one of our local newspapers, the Orange County Register, and a really interesting thing happened when they were writing up this work, where you can see that the headline here is Climate Invader, but when I was actually able to look at the proofs of the headline in the article before it was published, they had actually used the term Climate Evader, EVADER. And my first response was, no, when I was talking about this, I said invader, I didn't say evader. So my first response was just to correct the journalist on this. And so they corrected it and it was published accordingly. But then I started thinking about it more and thinking, well, what is the correct term here? Because the reason that these native species are shifting their ranges is at their leading edge, they have an opportunity to shift. Maybe they're not, their range isn't limited by harsh climate conditions anymore because the conditions are getting more favorable. But at their trailing edge, so for example, for these whelks, these predatory snails, their trailing edge down in Baja California actually has been contracting because it's getting too hot or too stressful for them there. So in some ways, their movement is just trying to escape climate change, right? They're evading climate change. And the work that we're doing really is very similar and dovetails with, and in some ways it's sort of a retelling of the work that has been done for decades, thinking about how to support climate-driven range shifts, and should we be providing assisted migration, assisted relocation, managed relocation for species that might be vulnerable to climate change. And so I think this is where this work on using frameworks from invasion biology to understand the impacts of range expansions can really contribute to this debate that is likely to increase in magnitude as we realize that some of these native species might need help to persist and that we might actually want to help them expand their ranges. And so, some of the questions are similar and in some cases slightly different, whether we are considering a species a climate invader or a climate evader. So I'm specifically talking about range expanding native species here. So with a climate invader, a species that is shifting its ranges beyond its historical range limit, we might ask, what are the impacts of this range shifting species in its novel expanded range? Are any of the impacted species that are in that expanded range vulnerable to extinction? So are we concerned about this range expansion? And ultimately, what are the costs and benefits of potentially even controlling that climate invasion? Do we want to control that range shift the way we would the spread of a non-native invasive species? And then on the other side, with climate evaders, is the target species, the species that we might be wanting to help expand its range, is it at risk of extinction without that facilitated relocation? What are the potential impacts of that target species in the areas of relocation? And ultimately, what are the costs and benefits of facilitating that climate evasion? So I'm kind of just leaving these as questions that we can discuss during the question and answer session, and also be interested to discuss them during the networking lunch as well. So to summarize what I've talked about today, the work that I've done and synthesized that others have done has shown that invasive species tend to respond less negatively to climate change than native species, that climate change more often minimizes the impacts of invasive species, but it does increase them in a certain number of cases that might be those most concerning cases. that the impacts of native range shifts can be similar to the impacts of non-native species invasions, leading us to potentially even call these range shifts or these range expansions climate invasions. And that frameworks from invasion biology might help us to understand these climate invasions. So in summary, I want to thank the funding sources of the work that I talked about today, the National Science Foundation, the Borchard Foundation. I want to acknowledge all my co-authors and collaborators in the work I talked about, and give you some ways to contact me. If you're not able to stay in chat today, please feel free to reach out to me if you'd like me to send you any of these papers, etc. I've given my email address, and I'm on Twitter as well. So with that, I will, and I also will invite you on Twitter to follow our new NSF project, which is the project looking at effects of those predatory whelks, and that's at Range Shift NSF. And with that, I am happy to answer any questions.
Great, thank you so much, Cascade. I have a whole list of things that I want to ask you, but I'm going to turn to our audience first. So we have a couple questions in the chat, and just a reminder to our audience that you can use the raised hand button. So you have a reactions button at the bottom of your Zoom screen. That one of the options, if you click the little up arrow, is a raised hand icon. If you can't find that, just put a note in the chat, and we can unmute you so you can ask your question. So Cascade, I'm going to start off with a question from Russ that was in the chat, which was essentially asking about whether the native range expansion work has included any accounting for, or have you thought about the impact of indigenous people's movement of species sort of between colonial time and now that might affect what we consider to be its native range?
That's a really good question. So I would say, for the purposes of the work that I've been doing, I've been considering historic ranges to be ranges, say, up until 1900, in some cases, maybe 1950. There's not always a really great accounting of where species range edges were and when. I would say probably even worse than the invaded species. It's, it's an issue that I've even come up with the non-native invasive species. Actually, the work that I showed you at the very last minute, we were debating what to consider the species, because there are some thoughts that maybe in the 1800s, some of those species we consider as native might have been brought. So in some cases, some of those native species might be considered naturalized species just sort of before our accounting. So yeah, oh my, right? That brings the oh my here, because it is something I'm really interested in. And actually, early on, I was really interested in the idea of biotic interchange and these sort of invasions that happened 3.5–4 million years ago, when like land bridges would be formed and biotas would come together. And I think it's a little hard to say that we know confidently where all these species have been when, but I think what we can say is that there's a lot we can learn about these different types of range changes, and trying to keep in mind the context of them.
Yeah, that was helpful. Thanks. And you made me think of a couple things while you were talking about that, mostly related to a session that we have on tap tomorrow, which is all about managed relocation. And one of the panelists in that session is going to be talking about what we can learn from paleoecology to help us inform managed relocation and decision makings there. And so I feel like there's some really interesting connections between what you just said and I think what we're going to hear tomorrow.
Totally. I'm excited for that.
Yeah, me too. We had another question from Kelly asking whether there are current examples where humans are starting to help with managed relocation. I don't know if you know of any particular examples in marine systems, perhaps. And then there's a second part of that question that I'll ask after.
Yeah, I'm sorry that I don't have any off the top of my head, but I believe that there have been some. I mean, I'm, you know, sort of intuitively thinking that we would probably find some examples from coral restoration, where maybe it might be unclear whether what we're doing is managed relocation or just restoration in areas where corals used to live. But I'm sorry that I can't give you any specific examples. I'm sure others on this know more about that, especially in terrestrial systems. So maybe they can throw some examples in the chat too.
Yeah, that would be wonderful. And I always think about the different contexts of the species we talk about for managed relocation as well, that, aquatic species have a little bit of an easier time perhaps moving around than terrestrial species might. Very much acknowledging my plant bias here.
Yeah, actually, it's really interesting in marine systems, because there are species that are super dispersive and then species that aren't. And so, the super dispersive species, like there's species that have larval stages that spend, 3, 4, even more months in the water, and so can get spread really far, really long distances, thousands of kilometers. And then you have species like actually the species that we're studying, which are more like, so I think of those dispersive species like plant species with wind dispersed seeds, or that might get picked up by animals and get dispersed far. But then the whelks that we're studying are species that lay egg cases, and then the larvae crawl away from the egg cases. And so long distance dispersal is actually really really difficult, and spread is really difficult for those species. And there are, quite a few others like kelp species that have that don't disperse very far, that sort of thing. So there are really kind of two modes, and the dispersive mode could really affect whether we felt like we needed to help with relocation, that sort of thing, and whether we would expect to see much spread at all.
Yeah. I want to shift our gears just a little bit. One thing that you mentioned earlier on in your talk, when you were talking about interactive effects of invasive species and environmental change, I forget exactly the statement you said, but it was essentially on the scales that we've studied them so far. And so can you talk a little bit about that caveat of sort of like what the effects of spatial or temporal scale, what impacts those have on the patterns that we've observed so far about the interactions between invasive species and climate change?
Yeah, do you remember when I talked about that? Because I remember actually having some thoughts went right as I was saying that, and now I'm trying to remember what thoughts I was having.
So you were talking about the meta-analysis, and you were talking about the fact that at local scales that invasive species seem to have larger impacts than global environmental change.
Yeah, actually, when I was saying that, I was thinking over temporal scales. I was thinking that over the amount of environmental change that we've studied so far, which would be sort of near term, is what I was thinking. And maybe also, I might have also mentioned it in response to, the disproportionate responses of native and invasive species. Actually, I can't remember, but in any case, I think the temporal scale is really important, because I think as a physiological ecologist, and so I'm always thinking, there are gradual changes until you hit a physiological threshold. And that's where, the stuff really hits the fan. And so I guess that's what I'm thinking of, that, we've been looking at sort of moderate changes in climate, but as we hit more and more of these extreme events, that's where we could see really drastic changes. And some of my experimental work is moving towards thinking about these extreme events, because they can lead to really kind of like regime shifts, as we've seen in coral reef systems. We've seen like heat waves causing massive bleaching of corals and huge shifts in coral reefs. And so, I imagine those kinds of things happen in terrestrial systems as well when you have like massive droughts or wildfires. So I guess that's what I was thinking of, was more like temporal changes.
Yeah, that makes a lot of sense. That's where my brain went to. I was just curious I was wondering what you were thinking when you said that.
I know. I wish I could remember too. Yeah.
We have a very practical question that I think is actually important when we're thinking about how to apply risk assessments to assessing neonative species and invasive species. And so how easy or difficult did you find it to perform the ICAT impact assessments? So that was from Liliana.
Well, so here's the thing. I think it's not terrible, actually. So it is a lot of wading through papers. I think it takes less training than you would think. And so we've been able to have a lot of students working on this. And I think it's actually a really good project for them to get involved in the literature and learn how to do literature searching. And so I found that with relatively small amount of training, undergraduates can search the literature, find papers on specific species, because it really does have to be quite specific to the species. And then assessing the impacts, maybe a little bit more work there. I think so. So from that, I think it doesn't require a ton of training. And with some species, you'll find there's a big range where you'll have some species where there'll be 1, 2, 3, 4 papers on a single species, and that's really quick, although your confidence might not be as high. And then you'll have species like, the invasive green crab that would probably take months for a single person to do that, because there's just so much known. Now, one way that that ICAT has, one of the conventions that they use is that they typically use the largest impact that's been published. And so there are ways that you could, you wouldn't necessarily have to wade through all of those papers if you found that a species had a level 4 or a level 5 impact, because then you'd figure, okay, I've already identified the strongest impact that this species has. And so you could kind of cut your losses and not have to spend so much time that way. So does that kind of give you an idea that it depends on how much data is available. It can be done with a minor amount of training. And the nice thing is that IUCN is making this database where they will have ICAT assessments for a lot of the well-known invasive species globally that they will be archiving. And so you'll be able to access those. One could potentially update those assessments, but you could also just use what they've put together.
Yeah, that was helpful. And so I'm trying to put myself in our practitioner shoes as well. And so it does mean that you have to have access to a research database of some sort in order to get access to the papers if they're not open access. And then, and then once you get over that hurdle, it seems like the rest of it is, not something that requires extensive training in order to implement that framework.
Yeah, but I also just think that there will be more and more people using it, and then hopefully we will share our assessments. Bethany's group has done an assessment for some species in the Northeast, and so you can start there and use what they've put together, and I think you'll find that really useful. Anyone working in aquatic systems on range expansions, check out our paper and you would have that to work from. So hopefully we can do this in a bit of a coordinated way and make our assessments accessible.
Yeah, that's great. Thank you. So there's a number of resources that folks have put into the chat. So to our audience, if you haven't been looking at the chat, take a look at links there. And I think I'm going to start the conversation that I think will probably persist into the lunch break with Cascade, because this is a sort of big, hairy question that could go in a lot of different directions, I think. So this is a question that Isabel posed, and I'm just going to read it to you, Cascade. It says, if range shifts / expansions in both native and invasive species have comparable impacts, as far as we know, on the ecosystems they are moving into, is it futile to resist and or try to manage any of this relocation?
Yeah, well, I think probably the answer there, a lot of you have more experience with, just because you've been involved in the management and control of invasive species, and I've done just a little bit of that, and so my guess is that some of the same rules with invasive species will apply as with range expanding species, that it takes less effort to prevent an invasion than it does to control it once it's already started. So for example, if you do have a species where you have a small population arrive in a localized area, if you think that you might want to control or prevent that invasion, it would make more sense to do so early on. That certain species will be easier to control than others, like species that don't spread as far, don't have as many babies, that kind of thing. But it's going to be interesting, because I don't know how much of these conversations are happening with the native range expanding species. I mean, I think most of us are watching out for the invasive species, but would we recognize a native range expander if it hasn't been put on watch lists, that sort of thing? If we saw it, would we be concerned? Should we be concerned? And I think that's where some of the really interesting questions arise.
So it feels like ending on a question and sort of additional work to be done is a great place to stop. So yeah, thank you, Cascade.
I'm excited to hear from others, and I hope some of you will come visit the discussion room, because I'm just really interested to see where other people's thinking is on this, and if anyone has experience with this, not just as a researcher.
Great. Well, that was the reminder, everyone, that we are going to take a short break now, and we will come back together at 12:30. So our hope was that these lunchtime discussions will be quite informal and just allow us to have some time to chat with each other about topics that we all find interesting. So we're going to have a series of breakout rooms that you will be able to add yourself to. And if you have trouble doing that, we'll have some of the moderators and facilitators around to help get you where you want to go. And so we'll have a lunch chat. So go grab your lunch wherever it is, bring it back at 12:30. And then after that, we'll have the early detection tools and techniques session, and then we'll have another break before the afternoon panel session. So thank you, Cascade. Thank you, everyone in the chat, for asking your questions and dropping links and providing perspective. We will be back in just 15 minutes. The main room, everybody. We're going to give everybody just a moment to get out of their breakout rooms and back into this main session before we get started with the early detection tools and techniques speaker session. So hang tight. Okay, it looks like our breakout rooms are all closed and everyone is back to our main session. Let me just verify that's true. Yes, okay, good, great. Well, welcome back, everybody. I hope that your lunch discussions were interesting and stimulating. I know I ate like half my lunch, so feel free to keep munching as we move into the next session. The next session that we're starting is early detection tools and techniques. And so we have a series of 4 speakers that are going to be talking about sort of up and coming programs or methods in terrestrial and aquatic ecosystems when it comes to EDRR. So with that, Annette, are you here? Perfect. Annette Evans is going to be our session moderator. So Annette, I will let you take it away.
Great, thanks, Jenica. So hi, everybody. Welcome to our first afternoon session and the first of our short talks on a focused topic. So as Jenica mentioned, this is a topic that's going to focus on early detection tools and techniques, and we have 4 amazing speakers today. So each of those speakers is going to get 10 minutes, and speakers, I'm going to give you a 1-minute verbal warning when you've got just 1 minute left, unless it looks like you're on your concluding slide. So just to try to keep us on time. And then after each of those 10-minute talks, there's going to be 5 minutes for Q&A. So if you're just joining us, if you're interested in asking a question of a speaker, you can either wait to use the raised hand function during the question answer 5-minute period, or you can type it in the chat and I will read that out or direct the speaker to that. So without further ado, I want to turn the microphone over to our first speaker, Ruminto, who's going to be talking about early detection of invasive species in the lower Hudson using conservation detection dogs.
Hi, everyone. All right. All right, so yeah, I'm Arden Blumenthal. I'm from the New York, New Jersey Trail Conference Conservation Dogs program at the at the New York, New Jersey Trail Conference, and I'm going to be talking about how we're using dogs in the early detection of invasive species in the lower Hudson. So the Trail Conference, a brief overview. We're a volunteer-powered organization that was established about 100 years ago to help build, maintain, and protect trails outside of New York City. But we also help to preserve land, advocate for access to open space, and increasingly steward the natural areas that our trails run through, and that includes the management of invasive species.
I'm sorry, just to interrupt, we can't see your slides.
Oh man, what's happening here?
Or at least I can't see your slides.
All right, did that work this time?
Yes, perfect.
Okay, great. Awesome. Well, so the Trail Conference is actually the host of the Lower Hudson PRISM, which stands for the Partnership for Regional Invasive Species Management. There are 8 of those in New York, and in our region, the Lower Hudson PRISM, we have over 50 partners that are made up of nonprofits, businesses, academic institutions, and even individuals that our main goal is to prevent the introduction, spread, and harmful impacts of invasive species. So the Lower Hudson PRISM prioritizes the early detection of emerging invasive species, and because we know that we're most successful when we catch species at the early in the invasion curve, as everyone here probably knows. But also, as everyone has probably dealt with, species are hard to detect in the early stages of their invasion. So the Trail Conference thought of a potential solution to what we are struggling with in Lower Hudson, and that is the use of conservation dogs. So the methodology was actually established in the 1890s. Their dogs were helping wildlife managers in New Zealand find native ground nesting birds. And since then, the field has grown a bit. So the 3 main areas of focus that conservation dogs are used for are ecological monitoring, wildlife crime prevention, and invasive species and detection and biosecurity. You might be wondering, why dogs? Well, the obvious is that, we're adding a different sensory modality, and dogs have an especially powerful sense of smell, but why not horses instead of dogs, right? So dogs are great because they are specific, so we can train them to find a specific species or even a specific individual, yet they're flexible. We're always training, and we can modify that training. They're agile and easy to transport, so we can pop them into our field vehicles, and they can navigate some pretty tough terrain. We can see results in real time as we're surveying, so there's no need to collect samples and wait for them to be identified in the lab. And they can be more efficient and accurate than human searchers, which is what we were most interested in. So our goal in the Lower Hudson was to have the dogs help us with our early detection capabilities. We wanted to cover larger areas more efficiently. We were hoping to find the leftover individuals after plants in particular were moved by our seasonal crews and volunteers. We were hoping to have unbiased searches. We have a lot of sites, infestation sites that we visit year after year, and we can get caught in a rut of thinking that we know exactly where those individuals are going to be found. So we wanted to better delineate sparse populations and find boundaries. And we wanted to improve our prevention capabilities in the Lower Hudson by, including the dogs in our program. We're increasing our education and outreach impact because dogs are so charismatic, one would argue, much more than an invasive plant. So we got really started in 2019 with a grant from New York State Department of Environmental Conservation, and our first target was the invasive shrub Scotch broom. We focused on Scotch broom because it was an emerging invasive species in our region. We had pretty easy to locate sites that we had been visiting year after year, and the plant is identifiable for the first handler who got started. And as you can see here, these two photos, there are Scotch broom in both of these photos, especially in the spring and summer when the vegetation is high. It can be extremely difficult to find visually. But these two are finds from our conservation dog, circled in red, and you might not even be able to see them even though they're circled. So we thought that our first season out, it was a success. Our first year, we found 2,257 plants with the help of the conservation dog. We were featured in over 100 publications, getting the word out about invasive species in our region, and we started adding more species to our repertoire. So this is the current list of species we have and currently do work on. Several invasive species that might be some hot button issues here today. We have also some native species that we've added, and then we have some species that we are interested in continuing work on, but we need more research, and some species that we have retired for reasons that I'll go over. So I guess because we have limited time here today, the real, the meaty question is the conservation dog methodology the right fit for detecting emerging invasive species where I am, wherever you are? So from what we've learned, the dogs can really be an asset. We can increase our search area with less effort. We have a sticky sage infestation that's about 80 acres. And the dogs can search, that and more in one season easily. We can improve our survey accuracy. So the dogs helped us confirm that 6 kudzu sites in our region were in fact eradicated this year. So it's just that double check to make us feel more secure that we are surveying, that what we're finding or not finding is what's really there or not there. they are unbiased surveyors, so we have found new infestations, new populations of plants with the help of the conservation dogs. They in places that we weren't trying to look, or for species that we weren't even trying to survey for in the moment. So we found a Scotch broom infestation while surveying for bobcat scad, and we found a new plant infestation hundreds of yards upstream and of where we had been searching. So those are the unexpected advantages of having a surveyor that's following their nose instead of their eyes. And of course, they're great ambassadors. So everyone loves to sign up for a demo or an outreach event that they know the dogs are going to be at. And those are just extra folks that we're capturing. We have learned several things though since we began the program, and this might help you narrow down how you might want to use this methodology in the future. So the dogs really are best involved when you're working at a site with low density of whatever you're looking for. And just because a species is emerging in your region doesn't mean that all the sites that you'll be working at will be low density. And we saw this when working with Scotch broom.
1 minute time check.
Thanks. There's some pretty robust seed banks, and even when plants are removed, you have to time those surveys right so that the scent isn't overwhelming for the dog that's going to be there. We also learned that utilizing the dogs in the best way requires great communication between the folks who are surveying and the folks who are managing. You want to know who is going to actually do the management. Just because you find something is the handler doing the management right then and there, or someone coming back. And when is management being done? This is especially important when we're thinking about using pesticides. The dogs don't have the same PPE that we do, so to limit exposure, that's something to consider. Some projects, like I mentioned, need more investment in research or just training time. The example of this would be oak wilt for us, since it's a fungus and you can't visually confirm it in the field. And arguably the most important and on this topic is that the status of the species that you might be working on will change over time. So we detect spotted lanternfly egg masses. We started in 2019, and it was a rapid response project then, but it is no longer that in our region. So thinking about the distribution of what species you're working on now might be like in the future, and if the conservation dog methodology will still be helpful for you years from now. And I'd just like to thank the folks who've supported our work. And that was a very high level overview. So I'm free to take any questions, and you can always shoot me an email.
Thank you so much. That was so interesting. So we do have a couple of questions in the chat. So the first one is asking about the time or cost requirements that there might be for establishing and maintaining a dog detection program.
Yep, that's going to be different depending on what scale you want to do the work. So the initial investment, there definitely requires an initial investment. The time it takes to train the dogs is less than the time it takes to train the handler, certainly. So if you're bringing on someone who already has training, that will cut down on the investment time. But if you're training someone from scratch, I've been shadowing under our handler for multiple years now. I didn't even have start handling my own dog for a year and a half into learning about the methodology. Not saying that's everyone's case, but you can train a dog in a couple months, a month or two, but to have a fully functioning dog handler team is a little bit more time consuming.
Sounds really intense. A follow-up question that some people had was asking, I guess more for clarification, that dogs can be trained to detect multiple species, or are dogs usually species specific?
Sorry, let me stop this. Yes, so we actually love to train the dogs on multiple species. Like I kind of alluded to, it can be an issue if the one species you've taught them is now everywhere. When it was initially an emerging invasive species or a rapid response project, it can be distracting when you're trying to look for another species, but this species that is abundant is they're going to be telling you about it everywhere they go. So we do love pairing multiple species together, though, especially native species projects. We think we'll be finding certain species in the same environments. It's just more data for the same effort.
Great. And then I think we have time for one last question, and that's whether your dogs are sourced from a particular breeding program, or can any dog be trained?
Yep. So a lot of other programs will rescue dogs. There are plenty of dogs that can do this work that don't, that there are more dogs that can do this work than there are handlers that are doing the work. And you just have to have the time and the bandwidth, the connections to screen for them in shelters. So or have the household that can maybe handle some dogs that have that come with a little bit of baggage. That's not every dog out there. Some shelter dogs are zero baggage. They just are fetch obsessed, and that's what we're looking for. So the dogs in our program are Labs and Malinois, and they're all from breeders. But we, the main thing is the dog physically capable of doing the work, and are they motivated to do the work? And most of that comes down to how much do they love their toy. So
Great.
We look for a fetch a ball obsessed dog.
Sounds like my dog does not fit the bill then. And so with that, I'd like to thank you so much. We do have to move on to our next speaker, but if anybody has additional questions, you can always pop them in the chat. And Arden, if you can see them, you can answer them or get in touch about them on your own time. So with that, we'd like to move on to our second speaker, who is called Tanya Bidden. She's from the New York State Hemlock Initiative. And she's going to be talking about utility of eDNA analyses for early detection and monitoring of hemlock woolly adelgid and biological control insects.
Hi, hold on one second while I figure out my screen share. I'm Tanya Bittner, and my slides are in the wrong place, so hold on. Let's go all the way back. Okay, I'll be telling you today about eDNA use for early detection and monitoring of the hemlock woolly adelgid and its biocontrol insects in Eastern North America. So first, let me introduce the characters in the story. We've got our antagonist, which is the hemlock woolly adelgid. It's a devastating pest in Eastern North America. Early detection could help save hemlock stands by getting them on track for treatment sooner. So our goal is to move toward putting eDNA in the toolkit for early detection. But we also are looking at some biocontrols. Silver flies in the genus Leucotoraxis are currently the focus of the federal strategic plan. Two similar looking species are imported from the Pacific Northwest, where they eat HWA on western hemlock. For these predators, fast and accurate monitoring would inform the overall biocontrol strategy against HWA. And to our knowledge, this is the first time that eDNA has been used to target biological control agents. But let's back up a sec. What is environmental DNA and why are we interested in using it? It's basically DNA that is shed into the environment through all the processes of daily life of the target organism. There's a huge body of literature showing that eDNA is everywhere and it's easy to obtain it. We target specific insects using quantitative PCR, which is very, very sensitive to small amounts of DNA, which also means that you have to be careful about contamination which I'm not going to talk too much about today. Now, another lab at Rutgers has published papers on insect pests, including the spotted lanternfly and the brown marmorated stink bug in forest and agricultural systems. And we've been working to design a simple, relatively inexpensive field sampling protocol based on washing small hemlock branches. This represents sort of the workflow, and let me talk just a little bit about qPCR. This little graph represents positive qPCR hits with different quantities of eDNA. So the x-axis here is time in cycles, and the ones that signal sooner, like this purple line, have more DNA in the sample than the ones that signal later, like this red line. So if there is no curve produced, that means that there was 0 target DNA in the sample. So that's just a teeny bit of background. Now this shows some of the materials that contain and transport the eDNA physically, maybe by wind, rain, or animal activity. HWA can be very abundant, as we saw in an earlier photo, those little waxy wool balls on the tree. HWA produces this waxy wool, as well as honeydew, shed exoskeletons, and lots of eggs, which all can contain eDNA. The predators are there as well if you've introduced them. If they're there, they're going to be leaving shed exoskeletons and larval frass. So this is just some of the source of eDNA that we can wash off of branches. So I'm going to have to breeze through this because these methods are standard and published. I can talk to anyone later that wants to connect if they want more information. But this is a vacuum filtration system. So it draws wash water through a small filter membrane at the base of a stand. We then use common practices to turn the gunk on the filter into a cleaned DNA sample, and then we do the qPCR. You can target multiple species in a single DNA sample from the environment, so that's pretty useful. And there are 2 ways that you can talk about your data. One is to simply do presence absence. So you say either it was present or it was not based on whether you got amplification. Another approach is to use a standard curve to estimate a count of how many copies of that little gene were present in the sample. So that's the quantitative part. And we might be able to learn more about patterns of spread by looking at quantities. So at this stage, we figure, why not do that? Last year, we published the assays and the proofs of concept in this paper in the journal Environmental DNA. For all the taxa that we looked at, the detection by eDNA was significantly greater than it was found by visual detection. The taxa that we used had to come from different sides of the country. So the HWA itself was studied here in New York, and the biocontrol predators were on foliage that came from Washington State. I'm only going to be talking about the HWA portion of this paper for you today. HWA has invaded vast areas of the state, but there are large areas in the north where early detection could still be useful. In particular, this treasured Adirondack Park has the highest density of eastern hemlock trees in the state. In our paper, we demonstrated the utility of eDNA for HWA detection on this little island called Dome Island. Within days of its first visual detection of HWA in October of 2020, we mobilized a sampling crew, which included tree climbing for a vertical component. 16 trees were sampled at 3 levels per tree. We then subdivided the 48 samples for eDNA and visual detection under a microscope. Well, unfortunately, we detected eDNA on all 16 trees that were sampled, but there was no difference in the quantity of eDNA between low, middle, and upper canopy samples. The copies ranged from just 3 to over 200,000 copies per gram of plant material. And visual detection was not very efficient. eDNA detected about 90% of the time. So what this says to me is that eDNA is highly mobile, and we don't need to go way up in the tree to find it. Moving on now to a more recent study. This one is not published yet. In 2022, we partnered with folks in the Adirondacks who collected a new set of samples in that same Lake George area and surrounding. And the goals for this were to investigate eDNA quantification on the edge of the pest's range and consider distance zones, by which I mean the distance from a known infestation like these fuchsia dots to a sample site, which are the black triangles. And I'm just going to focus on the core area results today. And these up here were negative, and these down here were very, very slightly positive. Here are the buffer zones, each with a 1 mile increment. And the dark dots that look red are the known infestations, and the other symbols denote our results, low, medium, high, or not detected. As you can see, there's not really a simple relationship between distance and detection.
1 minute time check.
Thank you. But we already know that HWA doesn't spread in a linear fashion, so that's not surprising. I think we can take away some evidence here that prevailing winds might carry eDNA. These no detection samples down here are very close to infestations, but they're in a spot that's sheltered from the wind, whereas these sites up here are in the path of prevailing winds. So it might be useful to consider winds as a factor in evaluating future detections. And we plan to follow up with visual surveys and repeat this entire process this coming year. Okay, now turning to predator detection quickly, we took a very large number of detailed samples at sites where we have released large numbers of flies in the past. In the initial screening, we screened for both species of flies. In the initial screening, we only saw a couple of weak signals for the species called Pinniperta. So we focused on quantifying the species Leucotraxus argenticalis. And so this is what we found. The 6 sites had a total of 69 positives. There were found in 5 of the 6 sites, on multiple trees per site, and on upper and lower sample grabs. The copy numbers were low, but that's somewhat to be expected when you're looking at small amounts of foliage. And sites that had positive detections, they all got a follow-up monitoring in the next year using foliage rearing containers and yellow prism sticky traps in the woods. Unfortunately, those efforts didn't yield any specimens, but it seems likely that flies had been there in 2021. But by 2022, the populations had faded out or had moved elsewhere. And I want to note that tree health at most of these sites had really deteriorated during that intervening year. So the summary, eDNA detection works for both pests and predators. It's highly sensitive, and we don't need to sample from high up in the canopy, which makes things easier. And future steps, we need to figure out the optimal sampling density, look at more sites, do it repeatedly, and follow up with visual surveys. We would like to learn more about how long the eDNA stays in the woods after it's been deposited. And we're looking towards possibly doing some other kinds of sampling that are a little more passive or might collect a larger quantity of DNA per sampling effort. With that, I'd like to thank everyone in our lab, as well as all of our wonderful collaborators, and happy to take any questions.
Great, thank you so much. So we probably have time for one question if anybody has something that they'd like to ask, either in the chat or using raise hand. Maybe just while we wait, you can always connect with Tanya after this. If you can just briefly speak to some of the issues of contamination. So what are some things that you do to try to minimize that?
The main thing is to collect a blank in the field as well as run blanks in the lab. So you're looking for any contamination on the field side of things and in the lab side of things. It's very easy to do lab blanks. Field blanks are a little more tricky, usually just opening a container and closing it again or something like that. But you have to keep all your field gear really clean because HWA, it just gets on everything. That wool is sticky and it sticks to everything.
Great, thank you so much. So again, if you do have questions that you think of, feel free to put them in the chat and you can reach Tanya through that way. But we're going to move on so that we can stay hopefully somewhat on time. So I'm excited to introduce our third speaker today, Sean Duffy, who's coming to us from Massachusetts Office of Coastal Zone Management, I think. And his talk is going to be on monitoring marine invasive species in New England.
Thank you very much. So yeah, I'd like to just quickly run through some of the invasive species monitoring initiatives that our office is a part of here in Massachusetts and regionally here in New England. So this is probably the last group I have to sort of have this introductory slide for, but I thought I'd just touch on marine invasive species, particularly in New England. So in New England, we've had transatlantic exchanges since colonial times and even before. So we have a lot of well-established invaders and more are coming, particularly in the marine and ocean environment. Many times these species are sort of out of sight, not of mind until they become a problem. And we often don't know the impacts of many of these species until they are a problem. So, you know, in the invasive species world, we have prevention, management, and eradication. All three of these are extremely difficult in the marine environment. You know, you have an open marine system. These species are pretty easily able to, you know, transfer and expand their ranges to different areas, primarily, again, sort of with shipping and vessel traffic. And we have one known example, to our knowledge, of successful eradication, which was Clerpa taxifolia in Southern California. And we currently do not have any sort of successful eradication stories here in New England that we're aware of. But we're always, you know, willing to sort of try new management techniques. So one of the programs that our office helps to coordinate in regards to invasive species monitoring is the Marine Invader Monitoring and Information Collaborative, or MIMIC program. This program started in 2006. CZM works with many organizations in the region, primarily nonprofits and research institutions, to train volunteers to monitor for invasive species at their local dock, pier, and rocky shore sites. So to date, we have hundreds of trained community scientists, volunteers, and we've monitored at about 139 sites from Rhode Island up through northern Maine. So in our MIMIC program, we have 18 established invasive species that we monitor for. We sort of have a taxonomic breakdown here of what those species are. The species were chosen primarily because they're relatively easy to identify in the field. You don't need a taxonomic expert. You don't need a microscope or any sort of other equipment to identify them. And all data, CZM sort of houses the data and does some QA, QC. And we have a Esri story map on our website that folks can use to sort of see the geospatial expansion of the species data. The second monitoring program CZM helps to coordinate with many partners is the Rapid Assessment Surveys, or RAS surveys. These surveys have been conducted since 2000. We covered New England as well as some sort of northern mid-Atlantic, New York, sometimes down into New Jersey. We get an expert taxonomic team together to monitor invertebrates and algae on docks and marinas. And we do collect data both on native and non-native species. So the RAS survey is conducted every few years. We try to get out every 3 to 5 years or so in the summertime, usually late July, early August. We collect physical and chemical data as well, including water temperature, salinity, and dissolved oxygen. And these surveys help to provide a preliminary baseline inventory of species, particularly in fouling communities. A couple caveats with the RAS survey, it is a small snapshot in time and space. We do have year-to-year variability in weather leading up to the RAS, which can impact the surveys and sort of what we're seeing out there. And the sites we visit can also vary based on sort of timing and funding constraints. So at this point, I just wanted to jump in quickly, sort of stay on brand and stay on theme here and talk about a few species that we've, through the RAS and the MIMIC program, have helped to sort of document both range expansion and initial introduction in the New England area. So both surveys help to provide information on range expansions and new introductions, which may be key as warmer ocean temperatures could result in an increase in successful introduction of species. So a couple examples here, we have Didenum vexillum, as well as Trisolaria inopinata. Didenum was first discovered in the Gulf of Maine in 1988, to our knowledge, and based on our MIMIC and RAS data, is now found relatively everywhere here in New England, certainly in the Gulf of Maine. So we've seen some pretty significant range expansion of that species. And Trisolaria was first discovered in 2010 at the Woods Hole Oceanographic Institute. And as you see here, found at every site but one in the 2018 Rapid Assessment Survey. And this included sites all the way up into the Portland area of Maine. So we've seen some pretty significant range expansion of this species in the last decade or so. The Rapid Assessment Survey can be particularly useful for identifying new introductions. And we have two examples here, Palameon elegans and Microdiopis grillotaupa. Palameon can be seen expanding its range based on our MIMIC data. We do have some indication from our Rapid Assessment Survey that there may be some declining trends in this species, but again, sort of the caveats with that Rapid Assessment Survey, small snapshot. So we're not fully convinced that the species is sort of decreasing in abundance in the area, but just sort of an interesting little factoid there. And again, here with Microdiopis, first recorded in 2003 in Buzzards Bay and Long Island Sound, and as recently as 2010, found as far north as Cascade Bay in Maine. And these species in particular are critically important for the Rapid Assessment Survey because many of these identifications, you need a specialized expert for identification. And again, getting back to that idea of, especially in the marine environment, some species are so small or difficult to identify that, many people may not even notice them until they're causing an issue, whether it's, economically or ecologically. Again, here we have another example of our MIMIC data helping to document range expansion. We have Guadalupe turatura, which is a red algae. first discovered in the mid 1990s in Rhode Island. In 2007, we saw expansion of the species into Boston Harbor at Rose Wharf. If anyone's familiar with the Boston area, that's one of our most frequently monitored sites. And in 2018, we saw expansion of the species north of Boston. And again, is now found as far north as the Damariscotta River estuary in Maine. Again, just showing, is it range expansion due to climate change? That's certainly a pretty strong working theory, but again, these monitoring efforts really help to sort of pin down when and where we're seeing these expansions. And I just wanted to throw in a quick example here that may be sort of counterintuitive. We have Colpominia peregrina, the sea potato, initially observed in Nova Scotia in the 1960s and has been expanding southward into Boston Harbor originally. And then in the 2013 Rapid Assessment Survey, it was observed at Sandwich Marina, which is down on Cape Cod. In 2013, we did have quite a cold spring. So, could that be how this species was expanding southward? Possibly. Again, our data here isn't high enough resolution to really tease out specific causes of these expansions. The data is really more useful for just documenting the expansion itself.
1 minute time check.
Great, thank you. So I'll quickly just wrap up. Introductions will continue to occur in the marine environment. Monitoring can provide early detection and information. And in terms of, management, our office, as well as many of the organizations we work with, continue to try to provide outreach and information on improved hull cleaning. We have one species of bryozoan in particular as an example that is continually found in New Bedford. To our knowledge, it hasn't established, but whenever we're out monitoring sort of after, the yachts and vessels are coming up from the Caribbean and other points south, coming back for the summer, we continually see this species. So improving hull cleaning practices on boats and vessels potentially could help sort of mitigate some of these impacts we're seeing. And lastly, I don't want to forget that although even in these highly altered environments, we are seeing significant marine invasive species, primarily the predominant species we find are still native, even in these highly altered environments. So again, on the Valentine's Day theme, I'll just throw this up there. We do find a lot of native species. We have some native amphipods, some sea grapes, some native tunicates, a Jonah crab, as well as some horseshoe crab. And this is work we do because we love our native species and want to help, facilitate their flourishing here in the New England region. So I will stop there, and I'm happy to take any questions if we have time.
Great, we do have time. So again, if you've got questions, either raise your hand or put them in the chat. So we have one question added to the chat from Ali, who asks, is there another RAS planned for the near future, since the last one was in 2018?
Yes, we are actually in the very preliminary stages of planning a rapid assessment survey for this coming summer. We bring in taxonomic experts from all over the world. We have some folks in Europe that typically come over for the survey, some folks from British Columbia and research institutions around the country that come in for about a week-long survey. So I'm happy, if folks are interested in either joining or just getting more information on that, I'm happy to put my contact information in the chat so folks can reach out.
Thank you. It sounds like a wonderful opportunity. This must be quite exciting. So while we wait to see if anybody out, there's a couple of comments in the chat mentioning things about examples of successful eradications in freshwater ecosystems, and also just talking about some of the challenges that we have in predicting the direction that some of these species may shift. And I was struck by your example, was it the sea potato that is shifting south? Do we have
moving south? Yep.
Yeah, like, do you have insights on like why? Since most things we think move north.
Yep, exactly. Yeah. I think the jury's still sort of out on what is causing that. Again, in 2013 in particular, when we found it on the rapid assessment survey, I think sort of that cold spring may have facilitated the movement southward, but in terms of the species being able to really establish long term, we're not really sure what physiologically is going on with the species to help it establish.
So weird.
Yeah, it's a weird one.
So we do have time if there are additional questions that people may have. Again, more comments for you in the chat of about native species that are moving south in recent years. It does. I'm assuming the survey is also tracking range shifting native species, since we've had discussions.
Yep.
Yeah.
Yep. I didn't have time for that, but we do have, blue crab, some other, sea bass. We do have some species that are, continually found on the survey. More mid-Atlantic, Southern Carolina species that are popping up more and more often every year that we're out there.
Great. Well, thank you so much for your wonderful presentation. And again, yeah, if people want to get in touch with you, it sounds like maybe putting contact details in the chat is the best approach.
Absolutely.
Great.
Thank you.
So with that, we're going to move on to our last talk for the session by Wesley Daniel from the USGS Wetland and Aquatic Research Center, who's going to be talking to us today about future aquatic invaders in the Northeast United States. So how climate change, human vectors, and natural history could bring southern and western species north. So take it away.
Thank you so much. Can you see my PowerPoint?
Yes, we can.
All right, wonderful. Thank you for the opportunity to present today. Today I'll be talking about a Northeast Climate Adaptation Science Center funded project where we're developing realistic future scenarios of expansion of southern and western aquatic invasive species, both native transplant and exotics, into Northeast water bodies. And we're basing this work on stakeholder input on the priority species. We actually let them decide the 100+ species we were going to model, and then thought critically about habitat suitability modeling, life history traits, known pathways, and projected all this into the future with future climate conditions in the region. So let me start off by talking about the project team and the other authors. A great group of USGS folks that are conducting this work, and unfortunately I can't spend much time on this. So the most effective and efficient way to deal with non-native and potentially invasive species is prevent them altogether into new water bodies. And the actions undertaken after invasion, of course, are more costly and less effective. But it's difficult to know which of the exotic and native transplant species could be invasive into a region. Many introductions of alien species fail or inconsequential to the structure and function of the ecosystem, but the potential to be invasive and cause impacts is essential information that can aid in the prevention or early detection of these species, and also prioritizing the allocation of scarce resources. That leads to our project. We're wanting to develop this kind of watch list of potential areas at high risk of invasion for already species that are in the US. And this is important because these aren't hypothetical species that could get here. These are species that are found in the Midwest or in the Southeast that have the potential to move into the region. And here's a couple examples from the Non-Indigenous Aquatic Species Database, which I coordinate. These are invaders on the doorstep, and you can see black carp, a known invasive Asiatic carp that's a molluscivore. It's moving up the Ohio River, and you also got a jump where somebody had transported it into some pay ponds as for snail control up into West Virginia. Or you can look down to alligator weed, a known plant that can move through transportation as a hitchhiker on boats and trailers, and it's creeping north as these maps show you. And so there's lots of pathways that these non-native species can move. Now, realistically, there's limited natural movement. You could, of course, move along coastal estuaries through freshwater lenses and kind of leapfrog up the coast. Organisms can move up the Ohio River or through the chain of the Great Lakes. But it's also critical to think about those human-mediated pathways. Are we moving water across these boundaries? Is there hitchhiking on our boats and trailers? Or are these organisms in trade or potentially going to be moved through fishermen wanting to make new fishing opportunities? So a very high-level view of the project as outlined. we're incorporating landscape data, thinking critically about hydrologic and climate and land use, along with some species that have been prioritized through our stakeholder involvement, and building out habitat suitability modelings of both current and future conditions. And then we're going to use these models to help kind of look at the suitability within the region and kind of score the risk of introduction through these multiple pathways, and the risk to individual water bodies within the region. We're going to use all this information and make it publicly accessible through interactive web applications, and of course through awareness through all the traditional academic means, manuscripts, reports, and presentations. So the products that we're going to develop include a priority list of invasive species, which has already been completed, a pathway of risk analysis, digital maps of both the current and future habitat suitability, and then those scores of those kind of targeted invasive species. And this is just an example of a terrestrial invasive species where you can see how future and current climate differ on their potential movement northward. So we developed our species list criteria through a vetting process where we look towards known native transplants and exotic species in the US, but that aren't tropical, because even in the face of future climate change, it's very unlikely that truly tropical species that might occur in South Florida or in Southern Texas would make it up to the Northeast. We limited it just to freshwater plants, invertebrates, and fishes. And we needed species with a significant number of occurrences, at least 100 occurrence records, just for the modeling practice. We developed about a list of about 129 species through 2 open stakeholder meetings. We shared the list and had vetting and some additions. And then we ended up at the end of the day with 110 total species. So greater than the initial proposal, but we're happy to add the additional species of interest. This represents 52 plants, 39 fishes, and 19 invertebrates, both mollusks and crustaceans. And I could give you a long list of those species with scientific names, which may not mean much to most of us, but here I'm just give you a couple examples. So on that list, we of course have some of the exotics that are either portionally in the region, maybe just in a couple of spots, or a risk to the region, like Eurasian ruff and zebra mussels. Of course, we also have important game species that are being moved around, like flathead capefish, is of course a native to the southeast. We have things that are moving through hitchhiking means, both with the water nymph, yellow floating heart, which is also to be found in the trade, and then of course bloody red shrimp. So a variety of species that are identified by our stakeholders. So right now we're in the modeling process, and there is opportunity for additional feedback on how we're doing this. And so we're looking for volunteers to review the models for ecological plausibility. And this is an important step in the process, because again, we all can make up hypothetical models of things moving around across the region and areas that have similar habitats. At the end of the day, we want this models to be useful for management, you know, and implied purposes. And so we're really seeking out volunteers to review these models for, you know, their actual usefulness. And so if there's interest, we have contacts here. First of all, we have our stakeholder group. So if you want to receive some future updates about this project, please contact Ian Finsting from USGS. He'd be happy to add you to our stakeholder group list and listserv and give you future updates. If you have any data you want to share, we're still in the modeling process and looking for landscape data, or if you just have questions, feel free to contact Ian or Peter Ingolstadt. And then if you do join our listserv, our next quarterly update is scheduled for April. That's going to be via email. And then we'll have a future virtual meeting when we have some results actually to share with everyone. So I see my time. I went much quicker than I expected, but that's okay. Here's all the contact information from all the researchers for this project, just in case you have additional information or any comments.
Wonderful, thank you so much. And yes, we have a lot of time for questions, and so feel free either to raise your hand or to put them in the chat. And maybe Wesley, if you can copy some of those links into the chat, maybe once the session's over, that way people can have the contact details in case they want to get in touch and involved.
Yeah, my pleasure.
You mentioned, I'm curious while we're waiting for some questions to come in. So you mentioned, the hope that once the project's done to share these maps and management resources, where would people need to go to find those?
I see two formats. We're going to make them publicly available through the Non-Indigenous Aquatic Species Database. So you could just come on and interact with the map on our webpage. We're also going to provide them out as a data product. So the GIS files on ScienceBase. And so if you had interest in integrating that data yourself and using it for other resources, that'd be available to you.
Great, that's wonderful.
Russ had a question, if you don't mind if I just.
Oh, yes, sorry. Yeah.
Okay, so Russ is asking, are there any native species, native further south and west, not but only limited spots in Northeast on your list? The answer is yes. Yes, we do have native species. Native transplants sometimes represent some of the more important non-native species that arrive. So for my database and how we track species, it's either native or it's not. And anything that's moved outside of what we think is historically the native range, we track within the database. So you can think towards red swamp crayfish, rusty crayfish, flathead catfish, blue catfish, lots of examples of things that are moving around that are problematic in the new ecosystems that they're introduced.
Wonderful. And I see Jenica Allen has her hand up. So Jenica, I'll turn it over to you.
Thanks. Thanks, Wes. That was a really interesting talk. Can you just talk a little bit about how potential impact plays into your framework? Whether you want to go back to a slide and I just missed it, or if you didn't talk about it, talk about how it's included or how it might be in the future.
So we didn't directly address impact within the model process itself. That's really addressed through the priority selection of species that we gave to the management groups. And so they were able to kind of tell us the species they're most concerned about, the ones that they see as being potentially or known impactful. So that's really how we kind of address that by itself. I think there's potential for any non-native species being introduced to be impactful, even if it's not measurable by our own means.
Great. So we have a question in the chat from Doug, who asks, for these modeling analyses, how will you quantify the magnitude of identified pathways and related propagal pressures?
So I love the magnitude question with the pathways, because that is an element we're still working on. There are many pathway analyses out there, and we're looking to see what's already been established. But I will be honest, I have this kind of idea in my mind of trying to think critically and something a little more quantitative than some of the qualitative pathway analyses out there. Doug, when I get there, I will let you know. It's something I'm working on. Now, propagal pressure, that's a little more challenging, because this is kind of a presence absence database that we're using, the data that we're getting from both my database and GBEF, the Global Bioinformation System. So it's really hard to kind of project propagal pressure, but I think the pathway analysis will look at credible movement in some of these pathways, and it could suggest risk associated with that. So frequency of boats in a lake, or proximity of a water body to a major metropolitan area would potentially represent higher risk of introductions versus a remote isolated water body.
That's really interesting. I was wondering whether so I'm familiar with the plant side of things in terms of these kind of modeling approaches, but notice you've got quite a few animals as well. So I know that there's some data out there on plant abundances. Is there similar data on the animal abundances that you use that in the future that could be incorporated into these models?
It could be. It's not the approach we're taking, but you're absolutely right. The modeling process that could be taken. The ensemble approach that we're using is kind of a presence absence approach. Abundance account for animals does exist. There are wonderful state and regional databases that track that. The hard part is getting that kind of information for a very large area. So those isolated data sets are extremely valuable, but are harder to model over large landscape scales.
Right. Last call for questions if anybody has questions. Otherwise, Wesley, if you can put your contact details in the chat, people can always reach out to you later on in this afternoon. But it seems like this is maybe a great time for us to move into, we're going to be having a 15-minute break following the session before hopefully you will all be returning for a session that is starting at 2:15. Jenica, is there anything that you need to share with everybody?
No, just a reminder that the session that begins at 2:15 is a panel discussion. So it's a building habitat resiliency panel. So it'll be a slate of folks who have been doing restoration, habitat restoration, and thinking about the climate resiliency piece. So yeah, come back at 2:15 for that.
Great. And I just had to say a big thanks to all of the speakers that we just had. And as Matt said, the next session is going to be wonderful. So we'll see you in 15 minutes.
All right, welcome back, everyone. Hope you had an enjoyable break. We're going to get straight into our next panel. The panel is titled Building Habitat Resiliency in a Changing Climate. Matt Brinka of New York Office of Parks is going to be our primary moderator. So Matt, are you here and ready to go?
I am.
All right, so I
Hear
will.
me okay?
I can hear you wonderfully. I will let you take it away.
Wonderful. All right. I do not have a camera on this, so you won't be able to see my wonderful face, but that's okay. So hello, everyone, and welcome. It's great to see so many people joining us. I hope you've been enjoying your time so far, but yes, I'm Matt Brinka, and it's my pleasure to be your moderation overlord for this session, and I'm really looking forward to it. Building habitat resiliency, especially in a changing climate, is a real interest of mine. So I'm going to take 2 seconds to talk about the session, and then we'll hop right in. So we have 4 people with us today, and we're going to start broad with a high-level overview of some of the intersections between climate change, invasive species, and policy development. And then we're going to get you out where you want to be, and that's in the field. We have some field guides with us, and they're going to provide a quick overview about multiple resiliency projects. And you're going to get to experience some beautiful grasslands, forests, and Great Lakes shorelines, and you're just going to probably have to plop on your imagination hat, I guess, to get the full experience. But we have a packed hour, and so each of our guides they have about 7 to 10 minutes to present. And we're going to hold all questions until the very end, where we're going to have about a 20 to 30-minute discussion. So feel free to slide your questions into the chat throughout the presentation so you don't forget them. And with that, I think we're ready to get moving. Sound good? So our first presenter is Molly Hassett. She is a climate forester with the Department of Environmental Conservation here in New York. She does the good work, the work that I don't really hope that I get too much into, but she spends her time and effort developing and implementing policy programs and guidance related to climate forestry and forest carbon. So Molly, do you want to take it away?
Yeah, definitely. Can you guys hear me?
Yes, we can. Thank you.
All right. So like Matt said, I'm going to do a high-level overview about climate change, invasive species, and New York State's Climate Act. So I really quickly want to do a quick background of the carbon cycle. So climate change, of course, is caused by atmospheric carbon dioxide and the greenhouse effect, by the excess atmospheric carbon dioxide from fossil fuel burning. How do plants and natural working lands fit into this? Well, plants sequester carbon through photosynthesis by trees, plants, and phytoplankton, and all of that carbon is stored inside those trees, inside those plants, and inside the soils. Carbon can move around inside the various like pools between the soils, the litter, maybe some of the animals, into the waters. So it's moving around. And that carbon that's stored can then be re-released back into the atmosphere through decay and fires and respiration. So these dynamics vary over time and over land use. So in New York State, forests sequester the highest amount of carbon per acre each year. Wetlands follow that, and grasslands follow that, and croplands actually emit a little bit of carbon. Wetlands and forests hold the highest carbon per acre inside their soils mostly, with wetlands actually holding the highest carbon per acre. So all these natural working lands are fairly important for different reasons on the landscape as well, and not just for carbon. However, talking about carbon and the Climate Act, carbon sequestration can help kind of offset carbon emissions that are released by fossil fuels from other sectors, such as transportation and buildings that are, burning fossil fuels. So that's kind of how natural working lands can fit into policy. So inside New York State, the landscape sequestration trends are driven by forests, and overall there has been a decreased sequestration over the past 20 years or more. Overall, that's driven by loss of forest land, the aging of forests because older forests sequester carbon more slowly, as well as forest pests and invasive species. So New York State has a Climate Act that has specific targets, including some of the energy targets you see there, 70% renewable energy by 2030, 100% zero emissions electricity generation by 2040, and an 85% reduction in greenhouse gases from 1990 levels by 2050. And there's also this goal here of 100% net zero emissions by 2050. And that goal specifically is where natural working lands come in, where that sequestration that those forests and those wetlands and those grasslands are taking in are meant to cancel out the remaining greenhouse gases that are left. So in New York, sequestration levels right now are at 27.7 million metric tons of carbon dioxide per year. And that, again, is driven by forests because New York State has a ton of forests, and forests are the best at sequestering carbon dioxide compared to other types of land use. With that 15%, I guess, kind of offset goal, that would be 60 million metric tons. So based on policy, there is a need to double the amount of sequestration. And for that, there was a scoping plan developed that was just released in December 2022, and there are recommendations inside that scoping plan. Some of the strategies and recommendations are to prioritize locations for natural resource protection, management, and restoration, keep forests as forests and prevent conversions, afforestation, reforestation, just planting trees, preventing invasive species and forest pests, promoting respiration, and supporting forest landowners and municipalities. So some invasive species-specific strategies that are inside the scoping plan is to increase the prevention of invasive species through strengthened regulations, inspection, and enforcement, as well as expanding the Department of Environmental Conservation's statutory authority to allow more rapid listing of invasive species for regulation. In New York State, we have a Part 575 legislation that process has to go through, as well as increasing the capacity for prevention, rapid response of forest pest management, and invasive species management. There's also some additional related strategies, and this doesn't list all of them, but some of them include restoration of degraded forest lands, as well as establishing trees on 1.7 million acres. So I wanted to also mention that it's not all just about the carbon necessarily. We also have to think about climate change vulnerability, including a future loss in productivity as well as regeneration success that will probably come with climate change. And that restoration will probably become more and more difficult. And it's really important to consider landscape resiliency, so having forests, having some grasslands, having wetlands, and encouraging just across the landscape a lot of diversity and vigor, as well as connectivity and function of all these different ecosystems. And there's my contact info. That's my presentation.
Thanks, Molly. And this kind of sets the stage. And, you know, just listening to your presentation reminds me that we all have our work cut out for us. And it just highlights a little bit on how tangled our ecosystems are with our individual state climate and carbon goals. And that, again, just highlights why restoration and resiliency play such an important role. So thank you again. And again, we're going to hold off questions until the end, but I hope you get to stay around. And we're gonna get out into the field. So I hope you packed your bags for a getaway, because it's time to travel, and we're going to go off to this far-off land that I've heard is named Nantucket, where Sarah Boyes, the Director of Research and Education for the Linda Noring Nature Foundation, she uses her passion for plant ecology and restoration to create resiliency in a globally rare sand plain grassland. So let's take a dive into some grasslands. You can take it away when you're ready, Sarah.
Great. Thank you so much, Matt. And I'm just assuming everyone can hear me just fine. Welcome, in this like travel theme, welcome to Nantucket Island. We're gonna, vary a little bit, being I'm in Massachusetts now, but I'm going to be talking about a project we've been working on managing some invasive species to restore our globally rare sandplain grassland habitat. So just to orient you a little bit where I am, I'm at the Linda Noring Nature Foundation, which is a land trust on Nantucket Island. We're on the western end of the island. We have 275 acres of open space. And this 275 acres has a lot of diverse habitats. We have wetlands and some small pockets of forest. We have vernal pools, we have shrublands, and we'll be talking a little bit more about our grassland and heathland habitats. We're open for passive recreation. And if you want to hear more about specifics of the property in our organization, you can go to llnf.org. But today I'm going to be talking about our sandplain grassland. So I know we talk a lot about non-native invasive species, but I'd like to lead with what are we protecting. So on Nantucket, we prioritize our sandplain grasslands. This map of the island on the right, the green is highlighting our grassland and heathland habitats. We sometimes talk about them together because they have a lot of species that are common. But the sandplain grasslands are considered a globally rare habitat. They're host to a lot of rare state listed species that we are mandated to protect. And while the plants make up the habitat definitions, they're host to a lot of rare Lepidoptera, as well as ground nesting bird species. So not only is it our value judgment and our management goals for my organization, but at the state level, we're kind of mandated to protect these habitats. And then just if you're interested in sandplain grasslands on a broader scale, we know that if in this map anyway, these reddish dots represent intact sandplain grassland habitats. And just kind of quickly looking at this, you can see that a lot of the intact sandplain grasslands, and in fact, a lot of the larger contiguous acreages occur on Martha's Vineyard and Nantucket Islands, with pockets in Long Island, and basically along this like North Atlantic coastal ecoregion. And so by protecting these remnant habitats in the areas that we manage, we're trying to figure out ways to protect and expand what we have. So I say this is a globally rare habitat. So first we know what are the threats, kind of knowing how to protect a species is knowing what the threats are. So over time, we have an understanding that main threats are habitat loss due to development fragmentation, natural ecological succession, so kind of succeeding into woody habitats, and then non-native invasive species, so new and existing threats. But I kind of parse out the different threats to also then look at all of the threats that we're seeing now with, the impacts of climate change. So further habitat loss in the area that I live in on Nantucket, directly to erosion, things like saltwater inundation through sea level rise and storm surge that we regularly experience now. And then additional climate impacts. So many of the things we've heard about already. So range shifts, both of our target species that we're managing for, as well as novel invasive species, maybe neonatives we've been talking about, other threats to these habitats. And so as part of my job being land management and making plans for our property, what are the threats that we can manage for? And so when we're increasing resiliency of our habitat, so protecting what we have in the face of change, what are the threats that I can reduce to increase that resiliency? I can't necessarily control a lot of these kind of bigger picture climate impacts, but I can try to control some of these other impacts. And so today we're focusing on obviously the invasive species component. So the species I'm focusing on our property is Pinus thunbergii, so Japanese black pine. It's kind of a scrubby pine. It is a non-native species. It's native to coastal Japan and parts of Korea. So it does occur in these nutrient-poor sandy soils on the coast. It has these little bundles of 2 needles. So it kind of looks like a native pitch pine to some people, but there's some easy identification characteristics. As you can see from this other photo, it can be a fire hazard. So as it gets to basically a certain adult size, it does grow pretty quickly, like a lot of invasives. It tends to die back because of some native pests. And then we end up with these stands like this, which are quite common in the area I manage, where there's a significant fuel load. And so a mix of standing live and standing dead and fallen dead as well. And originally planted as a windbreak, this is actually preventing some of the natural ecological processes of wind and salt spray that have shaped our island habitat. And then we also know that Japanese black pine is one of the potential hosts for the southern pine beetle, which is a southern native that is coming and becoming more of a pest species, primarily in Long Island, but moving into the coastal New England and Massachusetts. And then it also crowds out our native pitch pine. And pitch pine, Pinus rigida, is an important habitat, it being a native species, but also it's been shown on Nantucket to be a roost tree for the northern long-eared bat, which is just reached federal endangered species listing. So removing this Japanese black pine has multiple benefits. And this is once again our property. So the light green in this picture are the sandplain grassland and coastal heathlands. So you can see there's quite a bit on this one property we manage. And all the light blue, the different shapes, are actually pine, Japanese black pine individuals and populations. And so the triangles were just GPS individuals, or sometimes up to 10 trees in one area. And then the hash marked polygons are areas that we have actively managed to remove and encourage the sandplain grassland growth. And so just the dates on here just show that we've been working on this for a while. So since 2018, we've been managing different populations. And just a few months ago, or last month, we cut our biggest acreage up in 2023. So I have a series of before and afters, because it's always more fun to look at that than box plots, I think, for a management talk. But by cutting these individual trees, you can see from that pre-management picture on the top left, there are grass and forb species mixed in among the pines. And so our first step was to cut the trees down and restore the natural wind and salt spray regime. So trying to do as much with natural recovery as we could. The first years we did small acreages to test whether we needed to add any seed, or if native recovery happened within the seed bank and what was in the surrounding area. And so we've been really happy with that response. The immediacy, we reduced the fire risk, especially around trails that for people access, and also our office, which was right next to one of our bigger stands. That post-management photo is from the same exact spot, just during the growing season a year later. And for those of you who are curious at the management level about funding, we have received multiple different funding opportunities to conduct this work. So in Massachusetts, we were able to secure in 2 different cycles, 2 state wildlife habitat management grants. We had some private funding that was done in the name of grassland bird habitat restoration. So we had to collect some separate bird data for that. And then we also had some funding from the Regional Conservation Needs Grants Program through the Xeric Habitat Conservation Project, which looked specifically at native pollinators. And then just a few other before and afters from different parts of the property. You can kind of see on the left-hand side, this fall of 2018, you can see this little trail, and that is a good indicator of the same trail on the 2019 during the growing season. There are some bigger areas that were, there was a thick needle layer, so we used a lot of volunteers and student groups to kind of continue to manage those areas, but we were able to kind of have native recovery naturally. And once again, just
minute warning.
Sure, I'm just going to be done in 1 second,
Yep.
Or 1 minute, I should say. So you can see in this picture too, especially in the after photo, that Nantucket Sound is right up there. That's facing north. So you can see that north wind, which we experience in the winter, is really what shapes these low growing grassland habitats. So since 2018, when we started these projects, we've removed almost 20 acres of pine, but we say that's an effective restoration area of about 100 acres because of the wind and salt spray regimes that were restored to a larger area. We've had native habitat recovery of the sandplain grassland species and protection of native pitch pine stands. And then I have this picture of a lady slipper because we were noticing anecdotally and in some of our transects that we had over 100 lady slipper orchids come up in 2 of our different pine removal areas. Acidic soils, needle layers, and open canopy resulted in that. So those are unexpected positive consequences. And then the last slide I have is just mentioning that even though Japanese black pine was a known non-native, it wasn't on our invasive species list, prohibited list for Massachusetts, and it was still being sold at our nursery as a windbreak. And so in 2021, myself and one other colleague from Nantucket proposed 3 additional species, and we were able to get it listed. And so while it's not a widely planted species, it was important for that policy aspect to have another aspect to help further protect our habitats. And there's my contact information. If you have any specific questions about this project or anything else, I'm happy to answer anything.
Thank you, Sarah. You've made me basically want to frolic through a grassland and touch all the grasses with my fingers. So maybe not a globally rare grassland, but I definitely need to get into them more.
They like disturbance.
Okay. Oh, well, maybe I will run through them then. So we're going to take one of those rare direct flights from Nantucket to the Tug Hill Plateau in New York. And joining us is Robert Smith of the Nature Conservancy's SLELO Prism. Or for those who don't know what SLELO Prism is, the St. Lawrence Eastern Lake Ontario Partnership for Regional Invasive Species Management. That's a mouthful, so I would go with SLELO Prism. But Robert will provide a tale on how practitioners have been working to build multi-age climate resilient forests in one of the snowiest places in the Northeast. So Rob, take it away.
All right, thank you, Matt. Good afternoon, everybody. Like I said, my name is Robert Smith. I'd like to talk to you briefly about 2 climate change resilient forest restoration projects that SLELO Prism and the Nature Conservancy completed on the Tug Hill Plateau. For those not familiar with the Tug Hill Plateau, it's the 3rd largest forest landscape in New York, with a contiguous forest of approximately 150,000 acres. The plateau represents the westernmost portion of the northern Appalachian ecoregion and is critical to the Great Lakes ecoregion due to the high quality headwaters that originate on the plateau and then flow into Lake Ontario. It also has a history of timber harvesting. Much of this harvesting history has been in the form of high-grade logging, which means they cut down all of the trees of high value and then left the lower value quality trees. The result of this high grading is a forest that lacks diversity, has poor regeneration, a reduced tree species diversity, and it lacks the mature forest characteristics like snags and coarse woody debris that are very important to wildlife. This high grading has also left these forests less capable of adapting to a changing climate that is predicted to warm between about 4 and 6 degrees Fahrenheit by the 2050s, decrease in annual snowpack, and increase in extreme weather events like ice and windstorms. We also expect the arrival of new pests and pathogens that are well adapted to these new climate conditions and will be able to exploit trees stressed by climate change. The Nature Conservancy has owned land on the Tug Hill Plateau since 2002. When it purchased 45,000 acres, it kept 15,000 acres and then transferred the rest to the DEC with a conservation easement. In 2014, TNC purchased an additional 325 acres known as the Lindacre parcel. This parcel, much like other areas of the Tug Hill and the Northeast, was high graded. This occurred around 2009 and left a varied tree density and composition across the harvest area. Given the conditions of the Lindacre parcel, it was selected as an area in need of restoration. This gave rise to the Lindacre Parcel Climate Adapted Forest Restoration Project. This project and the next one I'm going to talk about were both funded in part by the Wildlife Conservation Society Climate Adaptation Grant. The purpose of this project was to test and demonstrate a climate adapted forest restoration project on the Tug Hill. The goals were to increase the amount of tree regeneration and to enhance/increase the species diversity. To meet these goals, it required a combination of managing the competing vegetation and planting of tree seedlings. This allowed the native trees to fill into these recently harvested areas before invasive species could establish. In 2017, the site was prepared by reducing the quantity of competing vegetation. This included beech bark disease trees, overstocked red maple saplings, and invasive plants such as common buckthorn and honeysuckle. As you can see down on the slide, 7 different treatment methods were used, and it was based on logistical factors such as budget and time constraints, as how we went about reducing these populations. In 2018, regeneration was accelerated by planting 35,300 bare root tree seedlings at a density of about 700 seedlings per acre on 49 acres of the Lindacre parcel. The seedling consisted of 14 different tree species and included both species that were underrepresented on the Tug Hill and those species anticipated to do well in the future climate. Climate adaptability of each of these species was based on the USDA New England and Northern New York Forest Ecosystem Vulnerability Assessment and Synthesis, which was written in 2018. Breakdown of these species can be seen on this diagram, with 7 species selected for being New York natives that are predicted to do well under future climate conditions, but are not currently on the Tug Hill. One species, black cherry, is predicted to be a climate change winner, and it's already present. And then the 6 on the right are species being selected to bolster the number of native species that are already there, but are predicted to do less well with climate change. In 2019, 35 1/30th acre plots were monitored for seedling survival across the 49 acres of planting area. Monitoring plots were selected to represent the variability in site conditions, species variability, and it was also based on accessibility. There was also a drought there to let you know that occurred in May-June period of that year, and there was no post-planting seedling care that was performed during this planting. In 2020, we found that 32% of the seedlings planted in 2019 had survived in the 35 plots being monitored. And this is lower than other plantings executed with bare root seedlings, which are usually about 50 to 60% after several years. Drought was thought to be a major factor in this, although other factors such as site preparation, sunlight, and variation in initial seedling size most likely also played a part. When species were grouped according to predicted adaptation, which you can see on the right here, to a low and a high greenhouse gas emission models, into a loser, winner, and a loser or winner with high emissions category, it was found that species considered climate winners had the highest survival rates at about 80% versus climate losers with about a 40% survival rate. Those considered drought tolerant were also found to have a high survival rate with 64% survival for high drought tolerant species versus 26% for the drought intolerant species. All right, next I'd like to talk about the second project that TNC started on the Tug Hill, which I'll refer to as the Tug Hill Patch Cut Project.
Hey,
It
Rob,
was implemented in 2018.
2 minutes. 2 minutes.
Almost done. Yes. It is located on TNC land just to the west of the Lindacre parcel. The purpose of this project was to increase the climate resilience of 2 even-aged northern hardwood forest stands. The goals were to establish multiple age classes in these forest stands and to promote tree species likely to do well under high climate change conditions. Management actions included creating patches in these 2 stands, planting tree seedlings, predicted to do well in climate change and change adaptable, and to increase the quantity of mature forest attributes such as snags and down woody debris. In 2018, these stands were prepared by harvesting 40 patches ranging in size from 0.4 to 1.6 acres in the 2 stands. Within each stand, at least 3 snags and 2 seed trees were retained, and also all the tree traps were left in the patches. In 2018, 8,527 tree seedlings were planted in 12 of these harvested patches to supplement the natural regeneration and increase density of climate adapted tree seedlings. The 4 species selected were red oak, white oak, sugar maple, and white pine. And climate adaptability of each of these, in this case, was based on the USDA Forest Service's Climate Change Atlas. In 2021, all tree seedlings were monitored for survival, height, and diameter by members of the University of Vermont. Results after 1 year showed that the red and white oak had a higher survival at about 75% compared to the white pine and sugar maple, with survival at around 50 to 60%. This study is very much still ongoing, so the reasons for the differences right now are being looked into and analyzed. Report should be available sometime following the analysis of this data. And that's all I have for today. Thank you for your time.
Thanks, Rob. You've reminded me just listening that forest restoration really doesn't stop with the planting of trees, and the monitoring and management of those is important. So I think it's a lesson many of us forget, but yeah. So we're going to switch gears, and I hope you packed a swimsuit because we have Dave Spearing here, and we're about to get wet. But we've hopped on a boat from the Tug Hill. Well, I guess you have to get to Lake Ontario first, down the Tug Hill, and we're meeting Dave, our Great Lakes Habitat Restoration Coordinator for State Parks. And he's going to step through some restoration efforts the agency is implementing to help build resiliency by restoring and enhancing nearshore fish and wildlife habitats, wetlands, and shorelines within the Great Lakes Basin. So, Dave, it's all you.
Great. Thanks, Matt. Thanks, everyone. Yeah, we are going to get a little wet here. So I'm going to talk about coastal wetlands and shoreline restoration. And before starting about the projects, I just want to put this work in the context of natural environmental variability that we experience. These are water levels across the Great Lakes for almost 100 years, and water levels go up and down. And that's a completely natural process that's going to continue. However, what we're seeing is new extremes. Lake Ontario set a new extreme high water level in 2017, only to be broken in 2019. However, this year, water levels are currently quite low in the Great Lakes. So really, what we're seeing is these new extremes that are unprecedented. The past has been the best predictor of the future, and it will continue to be, but we are going to see events that we have never experienced before. The picture on the left, I can speak firsthand. Here I'm in Niagara Falls right now, but I live in Buffalo. That Christmas blizzard this past year was like nothing I had ever experienced, and I lived almost my entire life in the Great Lakes. And I didn't have a great invasive species slide that captured the projects I was having that I'm talking to talk about, but this one I borrowed. And I don't think this is a group of people that doesn't realize how important invasive species will be in a changing climate future. So with that context in mind, I wanted to talk about 3 projects. So we're over on the Niagara River here. This is called Grand Island. We're at Beaver Island State Park at the southern end of Grand Island. Niagara Falls would be up in the upper left corner. It's Canada to the left of the screen and Buffalo to the right. And this project has a history that goes back before I was working on it. So in 2003, these offshore rock reefs were established to protect and help enhance coastal wetlands. And that needed protection from wave action. I showed those extreme pictures before, but that constant wave action, whether it being wind driven or boat wake, there's a lot of boat action on the Niagara River, created an unprecedented energy effect on the shorelines here and caused not only erosion, but deterioration of these coastal wetlands. So these rock reefs were installed in 2003, and they did a wonderful job of protecting this coastal wetland, and even we saw the expansion of it. So instead of just a fringe on the shoreline, we actually could see the wetland increasing out from the shoreline. So we thought that was a good idea. However, if you notice some vegetation on some of those rock reefs, this is what popped up. European black alder started to show up on these rock reefs. So when we expanded this project in 2019, we constructed 5 more offshore rock reefs to the south of the existing ones to expand and protect coastal wetland, because you can see the shoreline to the left there behind those new just stone rock reefs. We don't have that fringe of emergent wetland. So we want that established along that section of shoreline as well. We built these rock reefs at a lower elevation to prevent the establishment of that European black alder on them. And like every good restoration project, we heavily planted these projects. This is showing some of the nearshore plantings so that we can not only build on the existing wetland that was there, but really give a kickstart to that expansion from the shoreline we want to see. We use this first time for me using this different plant material, these planting bags. So imagine a sandbag that's pre-planted with 5 to 6 wetland plugs, and then you can see how easy they are to install. They're easy to move. It gives the plants a longer time, and they can be larger plants to grow in the nursery before we put them in the shoreline. So we really had great success with our shoreline plantings with these planting bags. However, out of the rock reefs, we also wanted to try and establish vegetation on the backside of those. And here you can see a higher water level day in the Niagara River with those rock reefs barely above the surface elevation of the water. Clearly a difficult place for European black alder to establish, but really it's been a difficult place for us to establish the other native wetland plants that we want to grow there. We have had poor success establishing plants on the backside of rock reefs, not only at this project, but in another one that we've worked on. And another invasive at this site is European frogbit. So luckily it's quite controlled at this site, or it's quite minimal in abundance. So it's been easy to control with hand pulling. But I brought it up here because this was not a minor component of the next project that I'll talk about. So this is at Buckhorn Island State Park. So now we've moved to the north end of Grand Island. And this project, we had a great remnant coastal wetland, the largest remnant wetland, coastal wetland in the Niagara River. And the issue here was being choked out with hybrid cattail. So we did a project to create channels and potholes. I think many people on this call would be familiar with channel and pothole projects because musky fish couldn't access this area for spawning. We started to lose bird habitat by losing the distribution of open water and emergent wetland. And again, I think this group of people on this call realizes why we've seen this cattleization, if that's a word, of the Great Lakes wetlands, water level regulation. The power lines at the bottom of this photo show, and they run to the hydroelectric plant operated on the Niagara River. Now those water level graphs that I showed earlier, some of those highs and lows are cut off. So the extremes have consequences in terms of high energy and shoreline erosion and loss of coastal wetland. But when we lose those extremes, we also have changed the conditions on how our native plants compete and occur. So now these channel and pothole projects have become quite common across the Great Lakes because of water level regulation and the cattleization of coastal wetlands. And here we had abundant frogbit. So we needed to go in there and control that. The open water gave us the ability to do that. Here's one of our happy aquatic invasive species technicians. This was probably early in the season because if you look at those numbers, he spent 900 person hours and pulled almost 20,000 pounds of frogbit out of there. We were only able to do that because if you look at the year 2020, that's the year COVID hit. That was, and our strike team could not travel throughout the state. Being outside was a safe place to be. So we would have not been able to give this effort to control frogbit at this level if we didn't have those COVID restrictions on other work they could have done. Luckily, we have not seen the frogbit come back in the abundance in the following 3 years that we've been continuing to monitor. And the last project I'll talk about here is now way over on the eastern side of the Great Lakes at Westcott Beach State Park on the east side of Lake Ontario. And here you can see a picture of high water levels in 2019, and that shoreline that's flooded was campsites. You can see on the left, there's the fire pits, the picnic tables, and we were losing that shoreline there. So parks took some efforts to increase shoreline resiliency here, and the first and the simplest and maybe the best, most effective measure we took is we retired those campsites. So now we no longer have the shoreline compaction and trampling, loss of vegetation that you'd get by having people camp and park there. So we took those campsites out of production and we installed the project that you see on the right with softer methods than stone. Those are coir logs, coconut fiber logs. Those will all be covered with sand, heavily planted with native vegetation. And now but when we see higher water levels in the Great Lakes again, this shoreline should be much more resilient to those higher water levels. And with that, here's my contact information, and please put questions in the chat, or we'll talk in a minute, or email me if you'd like.
Thanks, Dave. I don't know if I told you this. I'm originally from a small town along Lake Ontario, so your presentation was singing my heart song. But so we're gonna shift into the panel discussion. We have about 15 minutes, and then Tony Lynn had told me 3:15 hard cutoff. So there's more important things to get to, and I agree. So to keep things organized, you can either use the raise hand feature, so we can tell you to unmute yourself, or you can throw your comment into the chat if you haven't already. These are some restoration rock stars, and they're cornered. They can't escape. So ask those questions. And if it doesn't start organically, I am going to ask a question myself. So there is one. David, how are you accounting for increased erosion from recent increases in recent seawall construction?
How are we accounting for increased erosion from seawall construction? Is that the question?
Yeah, and I don't think that might be, yeah, that's the question.
Yeah, I don't know exactly what's meant by that, but I will mention a few of the challenges that come up from seawalls. So seawall, you have that vertical interface between the water and the land, and when you do have those dramatic waves, like one of the early pictures I showed of the Buffalo blizzard, or the blizzard in Buffalo at Christmas, that force has nowhere to go but either crash up, as you saw some there, but then also there's definitely wave energy going down. So you can have a scouring effect underneath the, at the base of that seawall. So that can start to undermine the integrity of it. Another big issue, and this wouldn't only be for seawalls, but for any hardened shoreline, there's littoral drift. So erosion is a natural process. We actually want erosion to happen. It moves into our waterways, it gets carried along to other places, and then it's deposited, natural beach nourishment, so to say. And when you harden a shoreline, you no longer are adding that sediment to the system. And when you start to think lake-wide, we've really started to starve some of our Great Lakes of that sediment. And that's on a big scale, but on a minor scale. And I've heard the word now used, erosion shadow. So the area immediately downdrift, littoral drift, when I'm saying downdrift, of a hardened shoreline, you can actually see increased erosion because you'll still have that wave energy and the scouring, the erosion effect at the high water and the storm event times, but you never will get the renourishment at the lower water or at the calm times because you don't have any sediment being provided from that adjacent shoreline that's now been hardened.
Dave?
Yep.
We have a question from Kristen O'Brien, and I'm going to change this a little bit, Kristen, but I'll keep this part of it there. So Kristen specifically is asking Sarah, how did you manage the needle layer with volunteers? But I think this opens up another question on a little broader sense of when we do our restoration, there's often follow-up management that needs to occur. And so I was wondering if each of you could provide just a brief idea of like what follow-up management should occur in each of the ecosystems that you're working in after your initial restoration has occurred. So Sarah, maybe we start with you since
Sure.
it was directed towards you about the needle layer.
Yeah, so when we first started removing the pines, we weren't exactly sure, what the grow back would be. And because we got a state grant for it, we had contractors come in to remove all the trees and chip them up and stuff. It was after we went through that money that we realized there was quite a thatch layer of needles. And so some of it was our contractors were able to do, but then we realized we needed some more action. And that was more of like an adaptive approach. We just kind of went as far as the money would go. So we did the thickest areas, we did raking and physical moving. Where the needles were more spread out, we just kind of left them and to decay. It was just, obviously that thick layer was physical barrier for things sprouting through. And so we've done that where we were able to. But you're right, for post like intensive management follow-up, we've been monitoring for, pioneer species that come in that maybe were unwanted. We have a lot of data to look at our transects for like, grow back of grassland and forb species. And that was, that's the monitoring is just as important as the follow-up management too.
Rob, you want to take 5 seconds to talk about follow-up management? You're muted. There you go.
Oh, go ahead. Say again.
You want to provide just a couple seconds on like follow-up management in restored forests, just so they don't become even-aged?
Oh, yes, yeah. There, the first example we had the Laker project was an example where you had high-graded clear-cut forest, and the restoration work that was done there, you know, it provided the greater increase in diversity there, you know, and increase in regenerations. But you have to follow up on projects like that, because what that doesn't have is it's when it grows up in the next couple of decades, it's going to grow up evenly. So you've got greater species diversity, but you still have age diversity, which is also crucial in resilience in these forests. So you have to follow up on these kind of forests. And the Lindacre one, I would recommend in maybe a couple of decades that they think about doing patch cuts like they did in the second example I gave during the presentation, where it was already an even age, maturing, you know, younger age stand, but still, you know, a stand that was ready for those patch cuts. So I would recommend doing something similar in follow-ups to, you know, projects like we did at Lindacre.
And I'm just going to follow up real quick with a question in the chat with you, Robert. Was there deer exclosures with your restoration?
There was some in the first ones, yeah, you have to forgive me because I wasn't there for the early parts of this. I don't have any significant data as far as whether that made a big impact up there at the tug or not, but there was.
Yeah, my understanding is the deer population in the tug, we still have a strong understory in that area of the state. So I think it would be a different story if we're talking about like the Finger Lakes, Hudson Valley, have severe browsing impacts, and that's always these side impacts to restoration. It's always about
No, absolutely.
eat up all those seedlings.
Yeah, absolutely. I think there you have that more contiguous forest there instead of the patchiness, and that's, you know, going to make it less favorable for the deer. So in other areas in New York where there's a lot of patchiness, that's really favorable conditions, habitat for deer, which makes restoration in those areas that much more difficult.
Dave, do you want to speak? I know you briefly touched on it with the black alder example. Do you want to talk anything about follow-up management to some of these like wetland restoration efforts?
Sure. I think Sarah said monitoring is just as important in the action as the action. And I think that's right. So you're going to need to monitor to know what you need to do. And we're trying to get better at that. That's hard. I don't think a lot of people, you know, have struggled with that as well. The grant is done, the money's on, a new project, you move on. But monitoring, and then the likely two things you're going to have to do are invasive species control. Hopefully that's minimal, and then often replanting. I'm a big proponent of overplanting on restoration projects because it's hard to establish plants and it's hard to give them a lot of the maintenance that they might need. So planning for some amount of replanting, and then what exactly you plant could be based on your monitoring.
April, your hand is up waiting patiently. Would you like to unmute yourself and ask your question?
Oh, thanks. I had a question for Robert Smith. with the sweet birch, I noticed in the one graph you showed that it wasn't one of the ones that was monitored. Maybe I didn't catch it. If you said it was monitored somewhere else, but I was just wondering if you were finding it to be aggressive. I work down on the Allegheny National Forest in Pennsylvania. We have a big problem with it and its aggressiveness. Thanks.
Goodness. Yeah, because there was a limited amount of plots that were monitored. I know there was a couple species that were not included because they just didn't make the plots. Yeah, we don't see I don't see a lot of over aggressiveness on the Tug Hill. Not to say that's not occurring in other areas, but not really on the Tug Hill.
Thank you.
I have a question here, and this might be a little contentious, so please panelists. But so I want to talk about the trade-offs between grasslands and forests, because obviously the management of grasslands hinders the succession into forests. So I was wondering if we could talk about the role of forest and grasslands, especially the interchange carbon resilience. So why don't we turn it to Molly first?
Yeah, well, I think it depends on what your goals are for the property. Obviously, Sarah works in an area that's globally rare, so I think it's really important to maintain some landscape diversity, especially since when you look at the landscape in the Northeast right now, there's a ton of mature forests out there and not as much young forests, not necessarily as much grassland, not as much old growth forest. So it's kind of a balance as far as like biodiversity and even climate resilience goes, because I think you need a diversity of all those different kinds of ecosystems to maintain climate resilience, which will help us, I guess, maintain carbon sequestration as well as carbon storage in the future.
Sounds like you're a component to grasslands. I was hoping there'd be like some interchange here, but Sarah, you wanna add on to that?
Yeah, I think obviously, like, because of my talk, I'm a grassland proponent in the sense of what I'm working with here and managing. I'm also a founder of the Sampling Grassland Network, so I feel I'm like, go grasslands. But I think that also, I don't have the paper off the top of my head, but there was some recent research that showed actually a lot of additional carbon sequestration in grasslands based on the underground biomass, so the root systems and things. So it might be higher than some people expect in general. But I think that it is, any management is a value judgment. You can have the science behind it that helps you guide your choices, but it's really up to what the mission of the organization or the goals of the project are and things like that. So, and it's also part of my, listing out some of the additional benefits of removing the tree species we were removing. It's to show that it's not just like we only care about grasslands and the species that are in there. It's that we're actually on Nantucket, because our climate is actually more closely related to Long Island than our mainland counterpart in Massachusetts. We're really concerned about southern pine beetle right now. We've had individuals detected. So we're, the bigger the island-wide conservation groups are doing a lot with our pine. Like this is like a separate project, but it's like a benefit that we're having by removing the Japanese black pine, because we're really wanting to protect our native pitch pine habitats too. And then it's looking not just at the species, but at that ecosystem process. So yes, we're limiting natural succession for these disturbance-loving habitats, but that's because they don't occur in very many other places. And so we're restoring that wind and salt spray regimes. And then, in other areas, there's, prescribed fire, there's mowing. We're not doing a lot of that currently, but it is all of that, monitoring, adaptive management. Are we reaching our management goals? And so go grasslands. It's a trade-off.
So you both have been talking about management goals and depends on what the management goals are on the site and so on and so on. So how do you balance competing management goals? I understand the rare species are rare standpoint, but talking, like, if you were to talk to one person and talk to another person, those have completely different idea of what a management goal should be at the site. And how do you balance that, especially in the light of climate change and invasive species exploiting? I want to take that a first stab at that, Sarah.
Yeah, just really quickly. So, aside from the rare species, we don't manage everything at once. So there's that diversity of management targets for different parts of the property, but then also timing is different. For something like our pine management, so Japanese black pines and non-native species, but because we're priority habitat in the state, it has to get approved by National Heritage. And so we have limitations on when we can remove the pines. So we can only manage pines in the wintertime, which is fine because they're evergreen. So, we have to manage them outside of bloom time for some of our rare forbs, but then also we have to avoid nesting time period for northern harriers. So sometimes the management that we'd like to do, like prescribed fire for a rare forb, is in conflict with the nesting period for a rare bird. So it's just timing and rotation, and that's how we kind of work with all those different things. Like anything with resiliency, the more variability you have, the more plasticity, the more biodiversity, the more varied you are, the more you can withstand all these different changes.
Anyone have a follow-up on that? I've thought this through a lot. Dave?
Well, I would say, Parks has to manage a lot of competing goals that aren't just natural resource related. We have a lot of recreation in our parks. So, but this is habitat restoration, and you'll hear someone say that, and it begs the question, well, habitat for what? Because you can't just say habitat restoration. So, like Molly was saying, there's a lot of mature forest, and that will be in the Northeast, that's the climax successional state that we will compete to. But now, since we have lost these disturbance regimes that create early successional forests, grasslands, all of those kind of things, we really have to have that intervention to go in there and do it. All these projects that, I showed, not all of them, but certainly the project at East River Marsh, historically, we wouldn't have needed to protect those coastal wetlands from extreme weather events, because around the other side of the island, it wouldn't have gotten the wave action the same way, and that coastal wetland wouldn't have been lost there. However, now, outside of the parks that bookend that Grand Island and the Niagara River, it's almost all private property, and now that's all protected shoreline. So we're left to protect the last remnants of what we have. So really, just like when an invasive species becomes super abundant, like buckthorn phragmites, we can't manage it everywhere. We have to pick our battles, and when we're losing something of real high significance, that's when we go in and try and control those super abundant invasive species, where we just, if it's already scrubby, second class woods, well, the buckthorn is just going to stay probably.
All right. Thank you so much. You guys are a wealth of information. So I need to wrap it up because we have a guest of honor. I do have homework for everyone, and it did come into as a comment, and I want to turn it around for everyone to think about. And I got a comment about how invasive species in general, not just invasive species, ignore boundaries. Whether it's land boundaries, political boundaries, or whatever it might be. So I want you to think about how you are coordinating with people adjacent to your properties, adjacent states, even adjacent countries, and find where those gaps are, because we all need to be together on these issues. So there's some homework for you. Reflect on your partnerships. And I will turn it over to Tony Lynn. And then I just want to say thank you to Dave, Molly, Sarah, and Robert for a wonderful session. And if you have any questions, I don't know if we're collecting questions or their contact information was in the slides and the chat, but if you need it, let me know, and we'll work from there. Wait, Tony.
Thanks, Matt. Such super practical information in that session. And so I think that's a great segue into thinking about how we do this work on the ground. So we are what Trevor Noah would say, like, brand new segment for our RISCC Symposium. We have a new award we're going to be giving out. So I'm just going to start by kind of putting us in context of where we're at. So here we are. You made it through the first day. We had this fantastic plenary and our networking lunch where we didn't really let you eat, or you had to talk and put food in your mouth at the same time so you can complain to us later. But I will say, if you say you didn't get enough networking time, we'll say, remember that time when we had you eat your food and talk at the same time? So there you go. And then we had these great quick talks and panel. So here we are now on our first Community Action Award that we're going to give out as RISCC across the whole network, actually, not just Northeast RISCC. We're instituting it right here. And I want to give you a little context on it. So some of you know that we sent out a questionnaire saying, what are you most worried about? What's the invasive that you're thinking about? And what do we want to tell the country about what we should be looking out for? And here's what you answered. So there's a lot of things, there's a lot of species in here, but I wanted to highlight one in particular, which is spotted lanternfly. By the way, if you want your answer to come up big, you have to make sure you spell it right. So just for future reference, you get extra points if you remember to put the words together right and get the spelling down. But you can see that this is a species that is of really big concern in our region. And you all are working hard to kind of get the message out that this is important for us to be working on. In fact, there's a lot of message out to the public about how should we deal with spotted lanternfly? They're so charismatic and obvious and such a problem that there is messaging out there. And this is actually from New Jersey saying, hey, if you see the spotted lanternfly, stomp it out. And so there's all this messaging to the public to do something about it. And so here we are today to celebrate a very important citizen action, a citizen that actually, community member who actually did some of that work to address the spotted lanternfly. And to introduce our award recipient, I'm going to hand things off to graduate student Ayodele Uhuru, who we were talking all about sleeper species today. So this is the brains and the brawn behind the sleeper species work. And so Ayodele is a PhD student in the Department of Environmental Conservation at University of Massachusetts and a longstanding member of the Northeast RISCC community. So I'll hand things over to Aya.
Thank you so much for that, Tony Lynn. Okay, so first I'd like to thank Bobby and her mom Monique for joining us today. Thank you. No problem. As a fellow New Jerseyan who also started my story basically in the backyard in actually Englewood, New Jersey, seeing aphids on a tree in my backyard, and basically turning that into my now story working with climate change and invasive species. It is an honor to present the first ever RISCC Community Action Award to Bobby Wilson. So on behalf of the Northeast Risk Management Network, we want to thank you for your hard work, your dedication, your resilience to seeing a threat in our ecosystem and choosing to take action to eradicate that problematic spotted lanternfly in your community. If everyone can, at this point, unmute yourselves and turn on your cameras to congratulate Bobby. We want to just say we want to continue to welcome, uplift, and highlight the youth that is leading the fight to combat the effects of climate change. So here you go.
Everybody wait, keep your video on. Justin, can you stop sharing so that we can see everybody? And we're gonna remove the spotlight so that everyone can be seen. All right, hold on. We're gonna give us one more. And then we're gonna, there we go.
Thank you all so much. Thank you. It really means a lot.
Yay.
And is everyone there in New York?
Yeah, so no,
everyone-
Monique, we're all over the place, and I forgot to mention exactly where you are. So let me get your spotlight back up.
Okay.
Yeah, so Bobby and Monique come from outside of Newark and Caldwell. Is that right? I got it right?
Caldwell, New Jersey.
Yes, yeah, outside of Newark, and
But not Newark.
Got it. And so we're all over the place. So we have people some of us are in Massachusetts, some of us are in New York, some of us are in Maine, some of us are in Canada, some of us are in the Congo. We're in Virginia. We're all over the place here to honor you.
The Congo, wow.
And
awesome.
Bobby, do you have anything you want to say?
Thank you.
You're welcome. We're so excited to have you as part of our community. And we're going to be sending your award and I think some insect guides in case that's something you want to do in the future. So you can ID things that you want to maybe keep out there, as well as the critters we're getting rid of that are problems.
Can you guys send enough for her classmates so she can do it with her classmates? Because they're all interested too.
That's an awesome idea. We can definitely send several.
All righty. Well, thank you guys so much. I want to get Bobby back into her math class, but I just want to say thank you again. We really appreciate this award. They all mean so much to us. And thank you again for honoring her. We really appreciate it.
Super to meet you. Take care.
Way to go.
Bye, Bobby. Bye-bye. Okay.
All right, everybody. So thank you for being game. And you know, we're all doing awesome work on the ground. So it was just like a fun thing we thought we could do, but you're all like doing such hard work out there. Thank you for spending your hours with us today. Come back here at the link, same link tomorrow at 11 o'clock, and we will have more presentations, more time for discussion, panels, conversation. Love how much action has been in the chat. I'll just say for the team that's running this, it's actually kind of a challenge to run an open meeting like this with 250 people and not as webinar style, but I think it's paid off because it's been all this conversation in the chat. So we love that. And yeah, everybody, happy Valentine's Day. However you celebrate it, remember your loved ones. There's somebody out there to send a virtual hug to or a real one. And we'll see y'all tomorrow.